Welcome, everybody. Thank you for being here. This is a very exciting time, and this is just terrific. It's been great getting to know some of the experts just before we get started here, and so I could not be more excited. We've got amazing people in the room. This FDA is an FDA interested in listening and learning and evolving, not just our thinking in regulation, but also our processes at the FDA. We are willing to challenge deeply held assumptions on how we do things if it means we can deliver more cures and meaningful treatments, safe and effective, to the American people. We have the greatest brand in the world, and we have to protect that brand and safeguard the public. At the same time, we have to create processes that are not in a receive-only mode, but partnering with developers and inventors so that we can reduce barriers and cut waste and get right to what's meaningful to the public. And so that's what we're here to do today. We're here to listen to you. The FDA should have a strong partnership with inventors and scientists and developers and companies, and at the same time preserve scientific independence in the product review process. And so we are deeply committed to that. Some people ask me, what's going to happen at the FDA when you take office? What are the changes? And specifically, people ask about the rare disease community, cutting-edge treatments, stem cell therapy, cell and gene therapy, many of the areas that are in your center, Dr. Prasad. And so that's been a source of a bunch of questions I've received. So let me be very clear. We are going to continue the successes of the FDA in facilitating the regulatory process for these conditions and for these products. We are also going to try and improve by creating more efficiencies. We are challenging deeply held assumptions in the traditional way that we do things. We are taking a look at the application process. We're asking ourselves, can certain materials be submitted in advance so that approvals can be quicker? We are asking ourselves, can we look at products in a way that a patient or the parent of a child looks at a product? And can we use post-approval monitoring and the assurance of post-approval monitoring in the actual decision-making process when we decide whether or not to approve a product? We want to continue to build on the successes of priority reviews, of accelerated pathways, of expanded access. We believe in both the spirit and the letter of right to try. So we are really here to learn from all of you today, and we're very fortunate to have a director of CBER who is medical and scientific genius, in my opinion, Dr. Prasad, who has written over 540 scientific peer-reviewed publications, and I think he's 15 years old or something like that. No, he's in his 40s, but he's a true medical genius who has been thinking about regulatory affairs for a long time. We've had many conversations before he began his job about the regulatory process, and I am fully confident in him and the fact that he is using gold standard science and common sense. And that's the theme of this FDA, gold standard science and common sense. So with that, I'll turn it over to Dr. Prasad. We have special guests coming today. We have the Secretary of Health and Human Services, the Administrator of the Center of Medicare and Medicaid Services. We have the Director of the NIH that will be joining us, assuming their schedules are not diverted with other national priorities that are things that they tend to, but we have an exciting day lined up. Thank you, all of you, for being here. Dr. Prasad.
Well, thank you so much. I should remember to turn on the microphone. Thank you so much, Dr. McCary, for that overly kind introduction. And indeed, we do have a number of guests coming later this morning to join us here and listen to your thoughts and interact after the program. Dr. McCary approached me when I joined, and we discussed putting this together, and the major purpose fits a theme of what we're trying to accomplish, which is transparency. We want to make, through our peer-reviewed publications, through our videotapes, through our podcasts, more transparent the thinking going on inside the FDA. We also want to be receptive to feedback, to thoughts that you all have on how we can do a better job of addressing the priorities and needs and scientific realities of your communities. So that's why we put this together. So we've invited 23 experts from around the country, and this is truly the creme de la creme. These are amazing biomedical experts we have here to discuss cell and gene therapy. And I have a few disclaimers to start. Of note, the personal opinions and views expressed by the panelists during this event do not reflect the US FDA's official position. Maybe not yet, but maybe we'll take some of them into account. Before we get started, the panel is being recorded and live streamed on YouTube and X, and it will be available in recording thereafter. Our panelists have microphones in front of them. Remember to push the button when you start speaking, as I just forgot to do, and turn it off when you're done. And we ask that the people in the audience, including the media, remain in listening-only mode and ensure that your cell phones and computers are silent. I just had a few quick remarks that I thought would interest you all. This week I gave a talk at the rare disease advocacy group, and I just hit 3 key points that I think we often get asked here at FDA. Number 1, what kind of therapies do you want? What are you looking for in therapies? Number 2, how will you make judgments as to the effectiveness and safety of therapies? And how will you engage with the community? What therapies we want? Of course, at FDA, we want what you all want, transformational therapies and cures and major advances. But we are also willing, and of course, will strongly consider any step forward that is in these conditions. We understand that progress is not always made in a single leap, and we will consider incremental steps forward because those add up. How will we make our judgments? We will rely on, as Dr. McCary says, gold standard science and common sense. So what does that mean practically? I'm often asked, what do you think about surrogate endpoints, which are endpoints that do not intrinsically matter to patients, but which often correlate with those that intrinsically matter? And what do you think about overall survival? How will you weigh these two things? And the answer is, we're interested in both. We will rapidly make available therapies at the first sign or promise of biomedical success or action, but we're also going to follow up overall survival and quality of life on the back end to ensure that we're accomplishing what we think we're accomplishing. I'm often asked, are you interested solely in randomized controlled trials, or are you open to other methodologies? And the answer is, we're interested in all of the above. Randomized controlled trials, whether or not people can serve as their own individual controls. We're interested in target trial emulation, inverse probability weighting, some of the more sophisticated epidemiologic study techniques. And I myself, in my laboratory in a prior life in epidemiology, have developed a couple of different trial designs, including something we call the iceberg plot, which compares outcomes for cancer patients on a novel therapy against prior therapies, and something we call a parachute trial, which is a way in which we protect patients from old, dilapidated, and antiquated control arms when we really have parachutes, interventions that efficacy is so demonstrable and visible. And I think some of you will talk about those things. So we are completely committed to bringing those into FDA and thinking about innovative trial designs in a way that hasn't been done before. Finally, I'm asked about trials versus real-world evidence, and the answer to that is both. We will always have a place for controlled clinical studies, but we will also leverage the incredible promise of real-world data, large evidence, and the methodologic techniques that are being explored in that space. And the last thing I'll say is, how will we engage with this community? As Dr. McCary says, today we're here to listen, but we're going to be very active in an engagement. We are doing a number of initiatives, including a listening tour where we're going to travel the country and listen to the CEOs of companies, big and small, to see what their concerns are. We've decided to try to open up what we metaphorically call the black box at the FDA. We're doing a series of video podcasts where, for our major decisions, Dr. McCary and I and others will discuss the rationale for those decisions and our thought process, including our recent COVID-19 framework, a video that came out yesterday. And we're going to engage in panels like this, talking to experts and the community more broadly. So those are my opening remarks. I'd like to welcome the 23 panelists here today. Instead of going through all your bios before, which will eat into the important time we have for you, maybe I'll call on you one by one, and then we will start to listen to your thoughts on this topic. The mandate is just cell and gene therapy, broadly considered what you think we ought to know here at FDA. So with that, let's get started. First up on my list is Terrence Flott. Hello, sir.
Great. Hello. Good morning, Commissioner McCary, Dr. Prasad, Dr. Verdun, and all of our colleagues at FDA. My name is Terry Flott. I am the Provost and Dean at the University of Massachusetts Chan Medical School, but I am here as the current President of the American Society of Gene and Cell Therapy, which represents 7,000 scientists, physicians, and patient advocates from academia, industry, and government. Over my entire career, I've been a physician scientist working on cell and gene therapies, and I'm honored to be here with you to discuss our opportunities to advance the field and patient access in particular to these transformative treatments. Thanks largely to a very productive engagement over the years with FDA CBER, gene therapy vectors have now been used successfully in humans for many different diseases, including approved products for spinal muscular atrophy type 1, SMA1, which is a severe, otherwise uniformly fatal infant onset neurologic disorder, IRD-RP65, which is a genetic form of blindness, as well as hemophilias A, B, Duchenne muscular dystrophy, metachromatic leukodystrophy, and others. But just as one particular example, the SMA1 product, Zolgensma, it has dramatically improved outcomes for infants with this disorder, such that treated infants are not only surviving, but acquiring the ability to sit, stand, and walk. These outcomes are completely unheard of in this cohort of patients untreated. As a pediatric subspecialist, I frequently encounter the parents of infants and children with rare life-threatening diseases who want to know why the technology used to treat SMA1 is not available to them and their children. Among the rare disease population, around half impact children. The U.S. alone has about 10 to 15 million pediatric rare disease patients, as you'll know from your visit at NORD. As you know, these diseases are individually rare, but collectively quite common. The most central strategic initiative adopted by ASGCT over the last 2 years has been that of access to gene therapy technology for all patients, including those with rare and ultra-rare diseases. We estimate that over 100 rare disease gene therapy products that had reached clinical stage have been discontinued since 2023, not because of treatment failure, but because of the risk of market failure. When such gene therapy products are deemed commercially pre-viable, academic centers are trying to step in to fill the void and continue offering these therapies. These considerations apply to both pediatric oncology and genetic diseases, the two disease types with the greatest number of gene therapy products in the pipeline. ASGCT's members appreciate the opportunity to work collaboratively with FDA to create a regulatory framework that encourages and supports the development and availability of CGT treatments for patients who have few to no other options. The agency's made great strides, however, there is more work to be done. Among the initiatives undertaken by CBER, the Platform Technology Designation Program may have the greatest potential to advance cell and gene therapy development by allowing a single technology, such as a particular AAV or lentivirus vector, to be used across multiple different disease applications with substantially less preclinical work prior to human use. We were very pleased to see just in the last day or two, the announcement of platform designation for Sarepta's RAV-RH74 to treat multiple forms of muscular dystrophy. What would be really transformative would be if in the future, designated platform technologies could also be made available to centers treating commercially pre-viable rare disease, in which this could have a dramatic impact. Similarly, the FDA-designated Advanced Manufacturing Technology Program for cell therapy could further streamline regulatory processes and de-risk investments in novel manufacturing methods so that we don't get locked into old technology. Other areas where the ASGCT recommends, commends the flexibility and recommends greater flexibility is in the areas of accelerated approval and innovative clinical trial designs, such as the Parachute trial that you mentioned that we've had an opportunity to read about, Dr. Prasad. We encourage providing greater weight to real-world evidence initiatives. As you said, real-world evidence can complement findings from controlled trials and enhance our overall understanding of gene therapy's impact on patients. Just a word, the FDA's Rare Disease Pediatric Priority Review Voucher Program, the PRV, provides incentives for life-saving innovations that might otherwise be commercially non-viable. The PRV program is unique in that it's a government incentive, but does not use government money. It's a win-win for product developers, patients, and their families, and it is very much needed to be renewed to attract investment in the space. So I'll just close by expressing again my thanks to the FDA leadership for putting this roundtable together. It's wonderful so early in the administration to do this in the spirit of collaboration. It's been my view that the most direct benefit to patients from the Human Genome Project have been within gene and cell therapy. And I believe that scientists and physicians all over have an obligation to do everything in our power to help patients and families living with genetic diseases. And that together, in partnership with FDA, we can bring innovation more quickly from the bench to the bedside. Thank you.
Thank you so much for those comments. I think, I'm not sure, should we comment on everyone? And then, no, we'll wait for the discussion at the end. We'll go through the panelists first. Next up on my list is, but we can switch the order because of how you're seated. Oh, you want to go, we'll go in the order on the list. We'll go in the seated order? Okay. Let's go with David Liu. He's next up in the seated order.
Thank you. Doctors McCary, Prasad, Verdun, and colleagues, I'm David Liu, a professor at the Broad Institute, Harvard University, and HHMI. And for disclosure, in the interest of transparency, co-founder of gene editing companies, including Beam Therapeutics and Prime Medicine. My laboratory developed base editing and prime editing. These are technologies that allow us to change a specified DNA sequence into a different sequence of our choosing. These technologies have entered at least 19 clinical trials. The results of 7 of these trials have been announced to date. And happily, in all 7 cases, the base editor or the prime editor located the target sequence, harnessing the beautiful DNA finding mechanism of CRISPR, efficiently made the intended edit using proteins engineered in our laboratory to perform chemistry on DNA directly, and benefited the patients as a result. The results from the most recent 2 of these trials have major implications that I think will be the subject of a lot of our discussion today. As you all heard, exactly 3 weeks ago, the first case of a baby receiving a custom gene-edited therapy was announced. Baby KJ Muldoon was born with a rare liver disease and faced a 50% chance of surviving infancy. A nationwide team led by Kiran Musunuru and Rebecca Ahrens-Nicholas that included our lab and several others at this meeting worked together to develop a custom base editing treatment for baby KJ in less than 7 months. Thanks to this treatment, today KJ is thriving, and I believe was on the Today Show this morning, in fact, while I was flying here. 4 days after KJ's story was published, another medical milestone was announced by scientists at Prime Medicine, the first treatment of a human patient with a prime-edited medicine, a second gene editing technology developed in our lab to make any kind of small DNA change, a substitution, an insertion, or a deletion. This patient is an 18-year-old that had chronic granulomatous disease, CGD. This is a primary immunodeficiency caused by a 2-letter deletion in this patient's case. Prime editing inserted the missing 2 letters, GT, into his hematopoietic stem cells, and after transplantation of these edited cells back into his bone marrow, restored the missing function in his immune system. A second patient has since been treated, achieving 90% correction of his hematopoietic stem cells. But just as Terry mentioned, as with most rare diseases, there aren't enough CGD patients to allow this drug to currently be commercially viable. FDA's priority review voucher mechanism that Terry already mentioned, during the short window remaining before it sunsets and hopefully is renewed, may provide a path to bring this treatment to more patients. Now, these medical achievements are not just scientific milestones. They also underscore a huge need and a historic opportunity for FDA, for HHS, and for this country. Each year, 10 million babies are born with one of about 10,000 known rare genetic diseases, many of which are in principle now treated with genetic medicines. The opportunity created by this perfect storm moment in scientific, medical, regulatory, and manufacturing innovation is to provide on-demand genetic treatments like KJ's at scale, making them the routine standard of care for life-threatening genetic diseases. With sufficient organization and federal support and partnership with FDA, I believe it will be possible by 2030 to treat at least 1,000 patients with personalized genetic treatments. Success would pave a path towards the ultimate goal, which is to provide each patient receiving a genetic disease diagnosis with a treatment made in time for their mutation to save their life. In addition to transforming patients' lives and those of their families, the successful treatment of 1,000 patients would save billions in costs that would otherwise be absorbed by our healthcare system just to manage their symptoms, rather than fixing the root cause of their disease. To achieve this goal, we need to build a national infrastructure for interventional genetics, including 1, rapid response diagnosis and therapeutic development centers to identify and optimize gene editing agents and their delivery vehicles. 2, small-scale, fast turnaround CMC capabilities. 3, cell, rodent, non-human primate, or virtual toxicology platforms. 4, streamlined regulatory pathways for N of few trials that do not, for example, require 3 PPQ runs at $7 million a run. And 5, shared public-private platforms for all of the above and for sharing clinical outcomes. Many of these elements exist already today and are ready for integration into pilot efforts within 1 to 2 years. This opportunity is not just about treating 1,000 patients, it's about building a modular system that turns rare disease treatment into a national engine for therapeutic innovation. Transforming personalized gene editing from bespoke science into scalable infrastructure would not only expand access to life-saving treatments, but would also generate major insights from treating patients that will catalyze future waves of therapeutic innovation, and importantly, would secure America's leadership in genetic medicine for the foreseeable future. Thank you.
Thank you so much for that, Dr. Liu. Just one quick follow-up question. So your prime gene editing, you perform apheresis, you collect the stem cells on someone ex vivo, you gene edit, and then you basically perform autologous stem cell transplant, reinfuse those cells?
In the case of CGD, the PM359 treatment, that's exactly what was done. It was ex vivo prime editing of HSCs followed by transplantation. However, there are ongoing efforts to do prime editing in vivo, both clinically and, of course, many examples in animals already published.
Wonderful, thank you. We'll talk more about that. Dr. Catherine Bollard.
Thank you. Thank you so much for inviting me here today. I'm Kath Bollard. I'm Professor of Pediatrics at GW and SVP and Chief Research Officer at Children's National here in Washington, D.C., where I also direct the Center for Cancer and Immunology Research. And I think it's also important to know I'm a bone marrow transplant physician who still sees and treats patients. And this is important because my patients really are always at the heart of everything I do. My conflicts are that I have patents in the cell and gene therapy space. I serve on the board of directors of Cabaletta Bio, the scientific advisory board for Manovia Therapeutics, and I serve as chair of the DSMB for SOBI. I always like to address where my accent is from early on when I speak because otherwise people just keep guessing. So I came to this country 25 years ago from New Zealand and naturalized to a new US citizen because I really did believe and still do believe in the promise of cell therapy, especially to cure children of cancer and immune-mediated disorders. And I came at a time when very few others believed in this promise. The first IND I submitted using a gene-engineered product was over 20 years ago, and over this time, my program and many other cell and gene therapy programs, including many led by my colleagues around the table, have treated hundreds of children and adults with cancer and life-threatening complications after transplant. Many of those patients, and in my case the majority, are children, are alive today because of the cell therapy products we developed in academia. One of the highlights of my cell therapy career was serving on the ODAC for the FDA that universally recommended approval of tisagenlecleucel that was developed from Carl June's group in academia to commercialization in a pediatric indication. Unfortunately, since that time, there has been no CAR T therapy that has achieved the same approval for a pediatric cancer indication, given that pediatric cancer is a rare disease and doesn't fit the traditional pharma model. So if we just focus on the children with solid tumors who have relapsed, we are still treating those patients with the same chemotherapy that we were giving 40 years ago. And yet we do have promising cell therapy options for these children with little to no industry interest in this patient population. So this large gap between academic development and eventual product approval for these rare diseases means that many of us are developing these extremely promising therapies and are currently left in this perpetual IND limbo, or what we like to call the valley of death. And so this is a problem that many of us in the field, led by my esteemed colleague, Dr. Crystal Mackel, are trying to solve by developing models that enables these products to be delivered in the advanced phase setting in academia. Obviously, patient safety and the quality of care when delivering these complex biologics is of paramount importance. And so to that end, I have one of my other hats on. As president, I also chair the board of directors in a volunteer capacity of the independent nonprofit organization FACT, or the Foundation for the Accreditation of Cell Therapy. So 30 years ago, physicians in our field recognized the curative potential of cell and gene therapies and the need to do everything possible that we are delivering quality, focused, patient safety treatments that will keep our patients safe. So shortly after FACT developed a voluntary accreditation process for all the facilities that provide these novel cell therapies, cell and gene therapies, to aspire to achieve, to maintain, and to further their commitment to patient safety when delivering these cell therapies. So if you like, our field already has this built-in safety mechanism in place, and these initial efforts have really thrived and expanded over the past 30 years. So we're currently helping bringing CAR T therapies closer to where qualified patients live. We're looking ahead to ensure our standards will also be accessible to cell therapies that are being developed for non-cancer diseases, with patient populations that are literally in the hundreds of thousands. So obviously this is really close to my heart to be in a leadership role with all the cell and gene therapy experts who make FACT what it is. So I really look forward to the panel discussion today, and to thank the agency for inviting us to the table, literally, to hear our perspectives and understanding. And I believe that we all share this common goal of developing and delivering cell and gene therapies to improve the lives of the patients we serve. Thank you.
Thank you, Dr. Ballard. You make a very good point about tisagenlecleucel, initial approval pediatric B-cell ALL, and since then we've had CAR T in follicular, in CLL, and diffuse large B-cell lymphoma, which are principally adult diseases, and we haven't had as much CAR T in pediatric diseases as we ought to, so we will take that fully under consideration. And speaking of, Crystal McCall, yes.
Good morning, and thank you, Kath, for the kind words, but thank you to the Commissioner and Drs. Prasad and Verdun and the rest of the FDA leadership for taking the time to meet with academic leaders and advocates as we're doing our best to move the field of cell and gene therapy forward for our patients. For the last decade, I've been a professor at Stanford University, where I've launched a Center for Cancer Cell Therapy. And before that, I spent almost 3 decades working in Building 10 in the intramural program at the NIH to develop cancer immunotherapies, and I had a major focus on pediatric cancers. Today, I am representing my academic community, as well as Access for Kids. It's a nonprofit that we've recently launched to develop and commercialize cell therapies for pediatric cancers. With regard to conflicts, I have founded 3 biotechnology companies, Lyle, Cargo, and Link Cell Therapies. I have many patents and I consult with many companies, but that work isn't relevant to my comments today. Because I'm focusing here on the pediatric issue. So when I look back at where we've come since the late 1980s when my journey started, the scientific advances that we have witnessed are just nothing short of spectacular. It's not hyperbole, it's not overstated. We could have never imagined at the time the power and precision that we can now endow into human immune cells, even human immune cells taken from patients with end-stage, heavily pretreated cancers. 1 dose of autologous gene-modified cells can cure about half of large B-cell lymphoma patients, about 1/3 of all patients with BALL, and it looks like 1/3 of now multiple myeloma patients whose diseases, in all those cases, were resistant to all other therapeutic classes. I mean, these are truly miraculous responses. We've been developing therapeutics for a pediatric brain tumor that is the largest killer of children due to brain cancer called diffuse midline glioma. And we're watching remarkable clinical benefit in these patients where they recover immune function that we thought was lost and irreversibly gone. Just this week, we had another 21-year-old patient who started the trial in a wheelchair and is now walking the halls of Lucille Packard Children's Hospital after infusion of these gene-modified cells into her CSF space. These treatments are being delivered with what we as scientists would consider first-generation versions of these therapies. And there are so many more innovations coming down the pike. And so we know that this innovation pipeline is just going to keep on giving, and we're going to have the opportunities for more and more novel therapies for patients who really have no curative options today. So this is all great, right? The glass is half full. But despite this unconditional scientific success, the field is really struggling to deliver these therapies to all patients who can benefit. I mean, even in the adult space, we only have market penetration at about 20%. But in pediatrics, we have essentially a complete market failure. As Kath mentioned, yes, and Dr. Prasad, we got Tisagen Leclucel in 2017. Not only have we not seen any other approvals come down the pike, what we know is in the developmental pipeline of biopharma today, there aren't pediatric indications there. It's just empty. Clinical trials, however, have demonstrated impressive results in pediatric brain tumors, solid cancers, neuroblastoma, T-cell leukemia, many of the biggest killers of children with cancer. But they're not moving beyond the research studies, and why? It's mainly due to the high costs of development and the regulatory risk. So enter things like Access for Kids and other nonprofits, where we're looking for ways to work efficiently and effectively to diminish the cost and relative and regulatory risk for developing therapeutics for these small markets, in my case, for pediatric cancer. And we all know that we need to uphold the quality standards, but we still have to make it somehow commercially viable or at least sustainable to get these drugs to market. So how can we work together? I'm hoping that we can work together and align on the evidentiary standards that we need to hit for rare disease to demonstrate efficacy, to agree and establish appropriate trial designs and endpoints for the earliest possible approval of safe and active therapies. We're hopeful that the FDA will endorse GMP manufacturing standards that are appropriate for small batch or small market manufacturing. These PPQ runs are just absolute barriers to entry. And that we can work together to figure out how to maximize the flexibility of the platform designations. I think it's great that this is moving forward for CRISPR. We need to figure out if we can use this for viral-based gene therapies. Finally, and very importantly, I'm learning so much about the economics of this. It's so critical that any marketing authorization that we get from the FDA is sufficient to obtain payment from CMS and the private payers. Fully 50% of children in America are on Medicaid, and if we can't get CMS reimbursement, we're really still in the same problem. I also want to highlight the critical importance of the priority review voucher. This is absolutely essential for any commercial, any business model that has any chance of being sustainable. So to summarize, we can wait for decades for scientific breakthroughs. That moves on its own timeline, and remarkably, we've seen them. But we can reduce many of the barriers we're talking about to access today just by working together and using what you say, gold standard science and common sense. Improve lives, save lives today, if we work together to find ways to develop and deliver these therapies at lower cost. Thank you so much.
Thank you so much. And I really want to echo one of the points you made early on, which is that it's just so impressive what CAR T cells can do when you collect even a multiply and heavily pretreated population of cells, and you can still turn it into such a potent therapy. And in terms of some of the audience members, we have some staffers from CMS and from HHS, and we have their bosses are coming soon. So I think that will be a way to talk about, I think, a very important issue slightly outside of FDA, which is coverage decisions. So thank you so much for those thoughts. Dr. June.
Well, good morning, Doctors McCary, Prasad, and Verdun. Thanks for this opportunity to speak today. By way of disclosure, I've co-founded several biotechs, spun out of Penn, and have numerous consulting agreements. And my comments reflect my personal perspectives and do not represent the views of the University of Pennsylvania. At Penn, I lead the Center for Cellular Immunotherapies at the Perelman School of Medicine and the Parker Institute for Cancer Immunotherapy. I helped pioneer CAR T-cell therapy. The CAR T treatment developed in my lab became Tisacel with Novartis, and the first gene therapy approved by the FDA in 2017 when Scott Gottlieb was commissioner. And thank you, Dr. Ballard, for sitting on that ODAC. Since then, over 50,000 patients have received this transformative therapy for adult and pediatric blood cancers. However, I have serious concerns about the future of cell and gene therapy in the United States. Despite our early leadership, much of the innovation in this field is now happening offshore, outside of FDA jurisdiction. If we do not modernize our regulatory approach, we risk losing our leadership and undermining the long-term viability of our biopharma industry. History shows that overcautious regulation can stifle progress. Bone marrow transplantation, where many of us started, was pioneered in the 1980s with only local IRB approvals outside of FDA oversight, and the field improved from initial mortality rates of over 20% to under 1% while improving efficacy. Had those high-risk early-stage trials been subject to today's FDA rules, they would likely have been halted, underscoring that rigid early oversight can hold back promising advances. In my view, the FDA need not regulate early-stage trials of autologous gene-modified cell therapies. Note here that I am not referring to allogeneic products given to many patients. I'm referring to bespoke, so-called N of 1 autologous products like hematopoietic stem cells, CAR T, and TIL. These exploratory studies can be responsibly run under local IRB supervision. I propose here a two-tier model, as China has, where first in-human regulatory exploratory trials proceed under IRB approval and full FDA local health review in China is required only once a therapy shows promise and is ready for larger multi-site trials. In China, investigator-initiated studies begin without upfront authorization from the national regulatory bodies. Only if a product proves promising do they move into the standard approval process, which closely then mirrors FDA processes. An increasingly common occurrence for me, and I'm sure my colleagues here around the table, is that we develop promising new strategies in our labs for cell and gene therapy, only to see them first tested clinically in China. Why are researchers increasingly taking trials overseas? Simply put, the U.S. process has become too slow, costly, and inflexible, while other countries make it easier to innovate. It's public knowledge that two biotechs, Interius and Capstan, spun out by my colleagues at Penn, are conducting their first-in-human trials overseas, even though they are backed by US-led venture capital. This is a problem and it urgently needs a solution. These differences have real consequences. In the past 2 years, over 100 CAR T-cell trials have begun for autoimmune disease, but they were launched in Germany with local IRB approval. We couldn't do it here. That wouldn't be feasible under current US rules, and unfortunately, the US has not led the development of CAR T for autoimmune disorders. Likewise, at this year's ASCO meetings, just this week, 2 major CAR T-cell trials were presented, one for multiple myeloma and the other for gastric cancers and other GI malignancies, and both therapies originated in China. These examples show that if we don't adapt, the next generation of treatments will emerge abroad. We are at a watershed moment. A wave of new therapies is coming from the innovation in academia and biopharma here in the US, but we can't test it in time. The stakes extend well beyond our initial individual trials. I worry about the health of our biopharma ecosystem. We saw what happened when investment dried up due to regulatory hurdles in the 1990s. Then my group collaborated with Cell Genesis to conduct the first CAR T-cell trials in cancer. However, biopharma concluded in 1999 that there wasn't a viable regulatory pathway to approval, and companies simply pulled out of the field and progress stalled for nearly a decade. I fear a similar retreat could happen today. Right now, cell and gene therapy startups struggle to raise capital, and many companies' valuations are below book. The path to approval is seen as so arduous. If firms feel there is no credible way to get new products approved here, they will simply relocate trials overseas or abandon them. We cannot afford that exodus. A regulatory environment that nurtures innovation and gives investors confidence is critical to keep industry and academia working together on US soil. Chinese momentum should be a wake-up call. In 2024, China surpassed the US in new drug clinical trials, with 7,100 in China versus 6,000 in the US, reflecting their streamlined regulatory environment. The US still leads in the basic biomedical science, but we must not become complacent. We must get a more enabling regulatory climate. In closing, I urge the FDA to implement regulatory reforms that will maintain US leadership in this field. Embracing a dual-tier approach for early-stage trials, like in China, will allow FDA and IRB-approved pilot trials to proceed, and involving only the FDA later in larger regulatory trials would maintain safety through local oversight while accelerating innovation. This approach would not lower standards, but would right-size them. So I believe this adaptive framework is essential for the US to preserve leadership in cell and gene therapy. Autologous T-cell trials should not require FDA oversight in initial exploratory trials. The future of medicine with cell and gene therapy is at stake. We must act now to modernize our regulatory pathways. Thank you very much for this opportunity.
Thank you very much, Dr. Jun. I think extremely important and salient points, and you're absolutely right. I was taking a look at CARTITUDE-1 5-year follow-up, and you see where it was conducted, and I think it's a commissioner's priority to bring those back to these shores, these clinical trials. So we will work with you on that. One matter of housekeeping, and of course a regulator has to do this. I've been notified that we're running at about 7 minutes a person, which is a little bit slower than we need to get done. So I have the unfortunate task as regulator to try to cut down the time to about 4 minutes. But that doesn't mean we're not very interested in talking here and then thereafter, right after this discussion. So with that said, let's turn to Donald Cohen.
Thank you very much for having me speak today. I think it's really great that you've gotten all these people together. So I'm a pediatric bone marrow transplant physician at UCLA, and I've spent nearly 40 years developing gene therapy for rare blood diseases, such as hemoglobinopathies, inborn errors of immunity, and metabolism. As my disclosure, I'm an inventor for an ADA SCID, bubble baby disease gene therapy that I'll be discussing today, and a co-founder and equity holder of Rarity Public Benefit Corporation, which is currently licensing it as our attempt to try to bring this to commercialization. One of the most successful gene therapy programs I've had the privilege to lead is for ADA SCID, an ultra-rare and fatal genetic immune deficiency. Since 2013, we've treated over 40 ADA SCID patients at UCLA using a lentiviral vector autologous stem cell transplant product. Our outcomes have been outstanding with 100% survival and greater than 95%, 39 out of 41, of patients achieving long-term restoration of immune function. The patients are thriving without medications, and families are again leading normal lives. Yet despite strong clinical data, the original licensee ended the program for commercial concerns before filing a BLA and returned the license to UCLA. In efforts to save this program from extinction, we founded Rarity PBC, a public benefit corporation, to lead the commercialization of this program. Over the past 3 years, since the return of the license and the IND back to us, we've worked to reopen the trial, secure grant funding, and build a viable regulatory and CMC development plan to BLA. The greatest barrier we face to date has not been scientific or clinical, but rather financial, and is primarily driven by the immense financial burden of meeting current CMC regulatory expectations. The transition from academic manufacturing to commercial scale production is estimated to cost between $25 and $60 million and take 3 to 4 years. This is untenable for an ultra-rare disease program that affects about 10 patients a year in the U.S. While the California Institute for Regenerative Medicine has supported and continues to support our program with grant funding, the current reliance on philanthropy is not sustainable or scalable. Academic medical centers have achieved remarkable efficacy in over 20 similar blood cell diseases, including sickle cell and beta thalassemia, using lentiviral vectors. But in order to obtain FDA approval, reimbursable therapies, we need regulatory flexibility that reflects the realities of developing treatments for ultra-rare diseases. Based on our experience, I'd like to propose 3 targeted suggestions for how the FDA might be able to right-size the CMC expectations for commercial approval, proportionate to the patient population size. First, can we permit continued use of GMP-like or IND-ready plasmids in BLA? The cost between a full GMP plasmid, and for vectors we need 4, at GMP grade might be $1 million each, whereas at GMP light it may be $200,000 to $300,000 each. So several $1 million difference just for the starting plasmids to make the vectors to make the cells. Second, can we reduce the burden of comparability studies transitioning from academic to commercial-based manufacturing? So many people have remarked on multiple PPQ runs, et cetera. For us, 1 PPQ batch of vector would treat probably every patient we face for the next 10 years. So to make 3 of those just to show that we can make it reproducibility, again, adds a multi-million-dollar burden. And finally, we propose an accelerated CMC pathway, similar to the existing accelerated approval pathway. In case where academic trials already provide robust evidence of clinical safety and efficacy, BLA approval could be granted based on core set of CMC data with post-approval commitments using real-world commercial batch data to support process characterization and validation. The FDA could also consider allowing the use of qualified assays without requiring full validation up front. These adjustments, if implemented, would reduce our developmental burden by more than half, from 30 to 60 million to 8 to 15 million, a large difference when you're trying to raise money from family groups, et cetera. Making commercialization feasible, encouraging sustainable models. So in closing, we urgently need a right-sized regulatory path for ultra-rare gene therapies, one that reflects the reality of clinical and manufacturing practices following academic settings. These therapies are working. Our patients are thriving. We just need a road to approval that's feasible for the diseases we aim to treat to make them sustainably available. Thank you.
Thank you so much for that. Starting to hear some recurring themes among the last 3-4 speakers, so thank you for that. Jamie Locke, Dr. Locke.
Good morning. Doctors McCary, Prasad, and Verdun, thank you so much for having me. It's a pleasure to be here. I am a transplant surgeon by training, and by way of disclosure, still have a clinical practice at NYU Langone Health. But I am speaking today on behalf of United Therapeutics as their Vice President for Medical Development for Xenotransplantation. You had asked us what should the FDA know, or what should be the emphasis. And I come to this both from the industry perspective, but also as the perspective of a transplant surgeon. And I would say that the thing that we must know and keep in mind is the patient's voice. I think it is critical to everything that we are doing, and I think we have to listen with the intent to understand. And I think that in many ways, what our patients want is simply what the Trump administration did during their first administration, which is they want the right to try. They want to feel that they have hope, that they've been afforded an opportunity. If I may, I'll speak to chronic kidney disease for just a moment. As a transplant surgeon, I'm well aware of this, but there are 37 million Americans who suffer from chronic kidney disease, and at any given time, about 500,000 or so will carry the diagnosis of kidney failure. We waitlist fewer than 100,000 of those patients, and this year, past year, even though we set a record in the number of transplants, we performed fewer than 28,000. And yet we know that with a transplant, patients achieve a significant survival benefit over dialysis. It gets them back to work, back to life. It restores hope. And so, without question, we have an unmitigated crisis in our organ shortage. I think it is something that President Trump recognized in his first administration when he issued the first ever executive order on a chronic disease being around chronic kidney disease, and actually encouraged the development of artificial kidneys. And I think our company, along with others, have really taken that to heart. And we've made great progress in the field of xenotransplantation, where we genetically edit pigs in the hopes of being able to use their kidneys and hearts and other organs to be able to treat patients with end organ disease. You've probably seen in the news many successes, and that is a direct reflection of our success with working with the FDA and the FDA's willingness to be nimble and issue compassionate use for us to be able to treat patients. I had the extraordinary privilege of transplanting one of those patients, TL, who kept her xenograft for 4 months and personally viewed it as an enormous success despite it having to be removed. And I bring that up because in her words, she characterized that as a stumbling block, but yet still saw it as a success, did not regret it, and encouraged us to continue and did not want that to be a setback for others. I'm pleased to announce that we do now have the first phase 1, 2, 3 clinical trial as a result of our efforts with the FDA, and there are other products coming online. But what I want to leave you with is that there is in fact urgency. About 20 patients die every day. That means in the course of our workday today, we'll lose another 6, 7, 8 patients. And yet we know what we need, which is more organs. And I think this field has the opportunity to provide that. And what I would encourage the FDA and others is to not hold xeno and these other therapeutics to a standard that is different than what we hold allotransplantation to. And remember the patient voice. They want that right to try. Every day in human-to-human transplantation, I have patients who sign up to take organs that have hepatitis C, that have all of these other risks, some known and some unknown, simply because they want the right to try, the right to live. And I would just encourage us to not hold these therapies to a different standard. Thank you.
Thank you so much for those comments. And I think even more broadly on the issue of organ availability, you'll find some of these HHS staffers I know have written extensively about it, and they're quite interested in it. So I think we continue this conversation even beyond xenotransplant, but more broadly as well. Dr. Ron Bartek.
Thank you. Thank you, Dr. McCary and Dr. Prasad and Dr. Verdun for, as always, listening to the patient voice at this important forum. My name is Ron Bartek. I'm the co-founder and president of the Friedreich Ataxia Research Alliance, or FARA, an organization that my wife Rachel and I founded in 1998 following the diagnosis of our then 11-year-old son, Keith. I'd like to say that FA, as I will call it henceforth, is a life-shortening recessive genetic neurodegenerative disorder that's terribly debilitating, and it's caused by an intronic mutation in a single gene that results in the reduction of the frataxin protein that is essential to life and to mitochondrial function. The good news here is we don't have to delete or replace that gene. We simply need to supplement it with a healthy copy of that gene, and we have every reason to believe that doing so would lead to profound benefit, especially if applied in these young lives at the earliest possible time. We have one gene therapy trial going in clinic right now, addressing the heart, which is our most condemning symptom. It's cardiomyopathy is our biggest killer. We have 3 others in late preclinical development targeting the CNS and the heart, and we also have 1 cell therapy program in preclinical development. I want to begin by expressing our gratitude for FDA and CBER and your wonderful Office of Therapeutic Products, especially in the following areas of real concern to us. First is for advancing accelerated approval so effectively. Second is advancing CGT with platform approaches in very rare disorders, and for collaborating so effectively with NCATS, the National Center for Advancing Translational Sciences, in so doing. Also to thank you for your efforts towards reducing CGT costs, a very important aspect that's already been mentioned by other speakers. For example, in manufacturing, if we can drive down the manufacturing costs, we can drive down the overall cost. Finally, I want to thank you both and for Dr. Verdun's tremendous, extraordinary team that you have there in the Office of Therapeutic Products and their extraordinary leadership in some of the most important issues to our community. One particular area of great importance to all of us is one in which Dr. Verdun and her team have been invaluable leaders, and that's in the inclusion of pediatric subjects as early as possible in clinical trials so that we can begin, as Dr. Verdun has said in the past, to prevent their symptoms rather than trying to treat them later. Get them into clinical trials early so that we can provide them access to safe, effective therapies at the best time for them, at the maximal point of benefit for them. Also want to note a few areas about which we continue to have concerns, and we feel the need for additional dialogue and clarity from the agency. One is that we think we need some additional dialogue regarding issues surrounding the difficulties with placebo-controlled clinical trials in scientifically plausible mechanisms like gene therapy. We'd like to get your help to consider comparison against high-quality regulatory grade natural history data, for example, and use of untreated subjects who could then cross over to treatment arm. Like everybody else in this arena, we're continuing to advance the use of AAVs and for delivery of our gene therapy and gene editing. We continue to pursue those AAV delivery mechanisms, but we're terribly concerned about safety, especially given recent tragedies. We wonder if there might come a time soon when appropriate, you could share with us some lessons learned in this regard, when it's no longer of proprietary concern, so that we could avoid making the same mistakes again. This is very important to all of us. We're also concerned about how to avoid beginning with doses too low, especially considering the inability to redose. Finally, I want to emphasize again the importance of early pediatric inclusion, and we formed actually a pediatric inclusion alliance to help socialize the powerful message we got from Dr. Verdun and the other centers in our July last year roundtable on this issue. And in that regard, I want to mention we paid a lot of attention last month to the FDA announcement that over the next 3 to 5 years, you'd like to make the use of animal models in preclinical data testing and collection the exception rather than the rule. And we want to make maximum use of the kinds of human cell-based technologies, especially those that are being now developed by our colleagues at NCATS, the human-based technologies, 3D bioprinting, human tissue chips, and so forth. And as we do so, we want to make a special request that we pay attention to collecting data from those human-based technologies that can report on pediatric inclusion. If we don't do that, we'll be stuck with even a worse problem. We can't even use pediatric animals to do. Finally, I want to ask for your continued support for the START program and the Rare Pediatric Disease Priority Review program, and conclude by saying our organization and the organization called the Alliance for Stronger FDA. We are very strong proponents and advocates for getting you the resources you need, especially given the fact that science is changing so quickly. You need to continue to have the leadership to help lead that science. So thank you very much.
Thank you so much for those comments, and I think you'll find that the Commissioner has been quite interested in this topic of minimizing animal research that doesn't provide information that can be leveraged for humans, so we're going to try to cut back on that. One more disclaimer before I go to you. I'm being told that we do have to watch the time and changing the time clock because we want to get to everyone a chance to speak before the Secretary gets here. Having said that, Terry Perovopoulos.
Yeah, thank you for the honor of attending this amazing, incredible session with people I deeply respect. I want to thank the amazing people at the FDA for saving and protecting our children. My story started on April 2nd of 2019 when my youngest son, Michael, was diagnosed with SPG50. We were told, go home and love him. There's nothing we can do. He'll be paralyzed from the waist down by the age of 10, quadriplegic by the age of 20. He'll never walk, he'll never talk, and it was a devastating experience for my family and I. We were fortunate enough to raise $4.5 million thanks to our community, find an amazing team. We liquidated our life savings initially and create a therapy for my son after an 18-month diagnostic odyssey. We found out what he had and then create a therapy for him and the other children. 90 days after he was treated, it took us about 3 years to create the therapy, but 90 days after that, we opened up a phase 1/2 in the US. We treated, since then, 8 children as of yesterday, and God willing, we'll treat more. We created Alpida Therapeutics after begging and pleading companies and institutions to take on this ultra-rare condition, but no one wanted to take this on because it was so significantly rare. We set up a social purpose corporation, run like a nonprofit. We've taken on 6 programs so far, treating 14 patients across 2 of those programs. We're also consulting for 6 other programs, and I'm helping personally another 100. Every day I receive 15 emails from families around the world pleading me to save their children. I've been offered, in the last 2 years, 40 programs dropped by biotechs from around the world. Everyone or almost everyone in this room has a career journey, except for me. But they do care and respect families and their journey. I'm here out of a nightmare that I will never wake up from. I put on a warrior's face over a broken man to save children because there's no one else that will do so. Because of my journey, I have a unique perspective. Being a parent of a rare disease, finding a team to make a therapy, making a mouse model, raising millions of dollars, filing and drafting a pre-IND, creating a therapy, filing and drafting the IND, treating my child, opening an phase 1/2, running a biotech, and now soon to be running a phase 3 study, working towards drug approval. Never in history have we been able to not only treat but cure our children. But funding and approval complexities are seeing that dream slip away from our children and our families. 95% of the other children that have no hope are told to go home and love their children. Nothing can be done. As a parent, I'm not asking, I'm begging Dr. Makary, Dr. Prasad, Dr. Verdun, and all the leadership to create a subcategory for ultra-rare conditions, for more flexibility when it comes to proof of concept and endpoints, allowing for a streamlined and accelerated approval pathway based on safety and the beginnings of efficacy, providing a lighter and faster package for ultra-rare BLA applications because several $1 million in 8 to 12 months is just not acceptable. Helping save not just the lucky 1, 2, 6, 8, 30 kids, but all the children out there that need to be saved. Let's not make this important meeting being a waste of air and words. Let's make and create policy changes. Think outside the box and help save these children. I pray every single child in the world does not have a rare disease, but pretend for a moment that your child has a rare disease and you're told that there's nothing that can get done. You find out that a gene therapy program or a therapy has been opened, treated 8 children and has been closed, and your child has now been diagnosed. And you're told that there's no more money to treat more children, and then approval needs to happen before your child to be treated. I ask of you to please think about that scenario and what you personally would do to save those children and your child. Again, I want to thank you for the honor of this, attending this event, and please, let's think of a way to move forward and save children like my son and the other 400 million Americans that are affected by rare disease. Or 400 million people around the world, I apologize.
Thank you, sir, for those comments. Paula Cannon.
Thank you. Hello, I'm Dr. Paula Cannon. I'm the immediate past president of the American Society of Gene and Cell Therapy and a distinguished professor at USC, where my lab is developing gene therapies for HIV. So as background, ASGCT is the largest non-profit professional membership organization devoted to cell and gene therapies, and we now have more than 6,400 members. It was established in 1996 by a group of the country's leading academic researchers, and even today that reflects the major role that academic research, and especially NIH-funded research in the United States, plays in this field. Our members are very focused on the approval of safe and effective cell and gene therapies and are available to consult with the FDA, and this includes an annual ASGCT FDA liaison meeting we hold. So it's very intentional that our society links gene and cell therapy together, because the advances in our field originated from the experience with cell transplantation therapies, notably bone marrow transplantations, and this led to early applications using genetically modified hematopoietic cells because of the ease with which these could be taken out of the body and engineered with the first generation of gene therapy tools. And this in turn led to treatments for previously untreatable diseases, starting with boy-in-the-bubble immune deficiencies and leading today to the extraordinary promise we are seeing for sickle cell disease. Engineered cell therapies have also given us the revolution of CAR T cells, which have changed the way we treat many blood cancers, offer promise for more insidious solid cancers, and are now also being used to target autoimmune diseases such as lupus. And at the same time, injectable in vivo gene therapies that have advanced to be sophisticated iterations of viral vectors and synthetic chemical mimics of viruses that can target tissues throughout the body and have also led to FDA-licensed treatments. And I have no doubt that the future will include the replacement of current ex vivo manufacture of modified cell therapies to in vivo engineering. And of course, as we've heard most recently, the extraordinary gene targeting capability of CRISPR-Cas9 and its derivatives, such as base editors, is offering the opportunity for highly personalized gene therapies and opening up possibilities for extremely rare, even N-of-1 diseases. At ASGCT's annual meeting, we heard about baby KJ, the first case where a child born with a rare genetic disease had custom CRISPR-based editors designed and packaged into a previously developed platform of lipid nanoparticles to create a personalized treatment that was given to the child within months of his birth. So this is a stunning example of the promise of these technologies, including the demonstration that the different components, the CRISPR genetic tool and the LNP delivery vehicle, can be combined in a sort of plug-and-play approach that could significantly reduce the burden of preclinical development for approval. So finally, a word about the need for speed in progress. We believe that cell and gene therapies are uniquely positioned to deliver safe, effective, and transformative therapies to patients who often have no other options, and for where doing nothing creates great harm. From the start, our field focused on safety, which was appropriate for these radically new types of therapies, but with our growing experience, and we've just heard that over 50,000 individuals have received CAR T-cell therapies, we are now in a position to put our foot on the gas pedal and consider innovative ways to facilitate the expansion of these treatments, especially in consideration of the needs of the rare disease population. Towards that goal, there is a pipeline with more than 4,400 gene, cell, and RNA therapies in development from preclinical through pre-registration. So approved cell and gene therapies sit at the top of a very big iceberg. And we recognize that these advances would not have been possible without the scientific and regulatory collaboration with FDA staff and CBER, and including the creation of the Office of Therapeutic Products, which has acknowledged the need for expanded capacity for future growth. We appreciate this support and look forward to new collaborations and innovations with FDA to support expanded access for patients. Thank you.
Thank you so much for that. And I think we're going to hear more about baby KJ later on in this panel. Timothy Hunt.
Great. Well, thanks. It's terrific to be here. Really appreciate the invitation. I'll try to make up a little bit of time, and I won't cover everything I initially was going to discuss. But maybe just a quick bit on my side. I think it's important to remember the past, the present, and the future when thinking about where we are with OTP today in the FDA. I started at ARM almost 3 years ago, and I heard constant grumbling about where we were with the FDA at that time. Good vision, poor execution, poor communication. Companies were very frustrated, and they walked with their feet. They'd pick up and they'd file the equivalent of INDs in New Zealand or the UK or Europe. I heard it all the time. The staff was well-intended but junior and did not understand the science. And that led to the creation of OTP, which was, brings us up to the present era, right, the last 2 years. Nicole Verdun and Rachel Anatol were brought in, and they became the reformers that really said, we have to change things. And execution became a huge watchword within OTP. Communication improved dramatically. The community, the broader academic, patient, biotech community really experienced an upswing in quality and a drive for excellence in science that I think has been incredibly notable. And we see it today. There's almost 1,000 cell and gene therapies in the clinic in the United States. There are about 30 approvals. The average life expectancy for these if you look at the natural history for areas where these gene therapies are approved, is 40 years. Right, it's about half the normal life expectancy. So these are devastating diseases where really time is life, and so a well-run, efficient, highly communicative OTP is really needed. And when you think about the future, I would just say, in our world, time is life. I have a son with a rare condition called Dandy-Walker syndrome. We're very close in our organization to the ultra-rare community. You've heard a lot, you'll hear more, and including with the pediatric community. Time is life in a lot of these indications, and I think, my ask would be, just as you guys I know are doing, continuing to think about how do we strengthen OTP, how do we ensure they have the necessary resources to get the job done. So with that, I'm gonna give you a minute and a half back.
Thank you, sir. Shengdar Sai.
Hi, I'd like to thank Dr. McCary, Dr. Prasad, and Dr. Verdun for the opportunity to talk. My name is Shendar Tsai, and I'm an associate member in the Department of Hematology at St. Jude Children's Hospital, as well as co-chair of the steering committee of the NIH Somatic Cell Genome Editing Consortium. I serve as a member of the Scientific Advisory Board of Prime Medicine and Insoma, although today I'm here speaking in my academic capacity at St. Jude. Our lab works broadly on genome editing technologies for therapeutics, including protein engineering to improve properties of these editors, translational efforts to advance genome editing medicines for pediatric patients at St. Jude for sickle cell disease, cancer immunotherapy, and towards personalized genome editing for bone marrow failure disorders. We also work on fundamental research and understanding the global activities of genome editors. Over the past decade, I've developed leading methods for understanding the genome-wide activity of genome editors, including the cell-based GuideSeq method, which was used to support the first commercially approved genome editing medicine, Exocell, as well as biochemical methods like ChangeSeq-BE, which was recently used to support IND clearance of the first personalized in vivo genome editing therapy with baby KJ. Our field is really at an amazing inflection point where genome editing could become a platform to treat genetic disease. And it's really amazing to see the progress from ex vivo cellular medicines to this recent report by doctors Kiran Musunuru, Rebecca Ahrens-Nichols, Fyodor Urnov, and team in treating this baby with a severe urea cycle disorder with a personalized genome editing therapy. We are currently working to push forward genome editing as a platform technology by really scaling up methods to understand the genome-wide activity of editors by several orders of magnitude. So this includes massively cellular profiling of the genome-wide activity of editors of thousands of targets, biochemical profiling of millions of mismatched targets. With these approaches, we and others are really beginning to train accurate artificial intelligence and machine learning models of activity. I understand that AI is an active area of interest at the FDA with some recent announcements and encourage the agency to consider, as these approaches mature, incorporating new AI approaches to assess risk in the future. I echo the comments of Terry and others, Carl, about streamlining of the regulatory landscape for platform genome editing, which would be very welcome. As you know, many companies are testing these strategies, for example, in New Zealand and the UK. We and others in the field envision this future of routine personalized genome editing, where there are use cases such as treating patients with many different mutations in the same disease-causing gene, correcting them back to wild type. It would be great to make the platform technology designations more flexible so that it doesn't start from a full drug approval, but could be available to more people. One other facet that would be really helpful would be rapid sharing of regulatory learnings back to the community. So clearly, there's a really thoughtful science-based approach at the FDA that learns from every IND and BLA submission. Is there a way where these kinds of learnings could be shared back more efficiently with the community? We're not looking for the agency to be prescriptive, but more examples of what would be acceptable responses to guidance would really accelerate progress so that we're not working separately and in the dark. Finally, I wanted to say that I'm really hopeful and optimistic about the future of our field. I started working in genome editing nearly 15 years ago at the intersection between zinc finger nucleases and talons, and we've really come a long way. We're poised to enter a future of hopefully many genome editing medicines, as David mentioned, and I hope that the agency can help us realize this here with a thoughtful and responsive regulatory environment here in the US that stimulates innovation and progress. Thank you.
Thank you so much. And your point about learning from the experience of others is well taken. The commissioner has a project on that he hopes to unveil soon. Charles Gersbach.
Thanks so much. My name is Charlie Gersbach, and thank you very much for the opportunity to participate in this very important roundtable. I'm a professor of biomedical engineering at Duke University and the director of the Center for Advanced Genomic Technologies there, where I oversee a research program focused on the development of new technologies for manipulation of genomes, epigenomes, and cells in a variety of ways, and using them for functional genomics as well as preclinical models of gene and cell therapy. With respect to disclosures, I'm a co-founder of two genetic medicines companies, Tune Therapeutics and Locus Biosciences, and also a consultant for Sarepta Therapeutics, as well as an inventor on patent applications and patents licensed to various biotech companies. So I've been attending the annual meeting of the ASGCT for the last 20 years, and I truly think that the meeting that we just had a few weeks ago was, for me, one of the most exciting and invigorating that I have been witness to. But I also think it has been somewhat mentioned in the earlier comments, brought into kind of starkly highlighted the dichotomy between the level of the science and the potential of that science and the overall broader enthusiasm in the biotech and investment communities. And so that's where I might focus some of my comments. I think that these early pioneering successes that the field has had for the last 5 to 10 years have shown the transformative potential of cell and gene therapy, as many of my colleagues have already discussed, whether it be curing certain malignancies with CAR T-cell therapy, the life-saving treatment of AAV gene therapy for spinal muscular atrophy, or things like the life-changing treatment of sickle cell disease with CRISPR-based gene editing. However, these very early successes of first-generation therapies also highlighted some of the challenges with this field, whether it be the cost of manufacturing and of customized, personalized cell therapies, issues with innate and adaptive immune responses to viral vectors, or difficulties in getting sufficient efficiency and biodistribution of tissues outside of the liver and or in older patient populations, and the list goes on. I'm very concerned that some of the pessimism regarding these challenges of first-generation therapies is drowning out the incredible scientific advances in these next-generation therapies that are directly overcoming some of these obstacles, as was highlighted at this recent ASGCT meeting, including things like in vivo cell engineering for CAR T and sickle cell disease and other indications, next-generation viral capsids that evade pre-existing immunity or increase the efficiency of delivery by orders of magnitude over the first-generation vectors, and next-generation editing technologies that can create whole new ways to treat both genetic and common chronic disease. And so what's been lost is that we're still at the nascency of this field, and just at the beginning, and these initial products are the earliest stages and not the end. And so we need to establish a plan to move these new technologies efficiently and economically forward, to realize the potential that was laid with the foundation of CAR T gene replacement and CRISPR gene editing. And then lastly, a related note to a personal interest of mine is how we move from gene and cell therapy for these rare genetic conditions into common chronic disorders. And so, for example, while CRISPR is famous for DNA editing, I think it's actually a misconception that that's why it's transformative. As was mentioned earlier, the transformative nature of CRISPR's ability to target any site in the 3 billion base pairs of our genome. Once you do so, there's a variety of other operations that you can perform besides cutting DNA or changing underlying DNA sequence. And in fact, it's other aspects of our genome, like our epigenetic regulation, that are responsible for common and more chronic disorders. And so I think that's why we're very excited about the newest wave of editing technologies to move into clinical trials that leave DNA sequence alone and actually manipulate DNA epigenetic regulation for more common disorders. And so I think that's just one example of many of where these new technologies can help us move from a foundation in rare genetic conditions to more common chronic disease. Thank you.
Thank you for that excellent point. Andrew Cameron.
Good morning, all. My name is Andrew Cameron, and I'm the Chief of Surgery at Johns Hopkins Hospital in Baltimore. I am here this morning not on behalf of the university or even the Department of Surgery, but simply as a physician scientist to discuss issues of relevance to our patients. I sadly have no financial relationships to disclose, but our laboratory does receive federal and industry funding to support our research in xenotransplantation, which you've heard mentioned and to which I'll return in a moment. The Hopkins medical system, of which I'm a part, actually consists of 7 hospitals, 2 of which are here in the National Capital Region, civilian suburban hospitals. And we have many issues of relevance at Hopkins related to gene and cell therapy, be it CAR-T or gene therapy for sickle cell disease. Of particular interest to the Department of Surgery is xenotransplantation. I'm a liver transplant surgeon by training, still in active practice, and there is a public health crisis in the United States in which well over 100,000 Americans currently wait for a life-saving transplant, many of whom will die waiting. That has proved a refractory public health problem. Most of those 100,000 wait for a kidney, but others wait for a heart or liver or lungs for which there is no dialysis. Kidney transplant is, in fact, one of the major accomplishments of modern medicine over the last 50 years, besides being a replacement for dialysis, which itself is life-saving, but absolutely miserable and quite morbid. Transplant is a money saver for the system. We could imagine 2 identical twins, both on dialysis. One gets a kidney transplant, the other remains untransplanted. The twin who gets the transplant will live an extra 13 years, and the cost of the transplant will pay for itself after 2 years off HD. It's not just extra years, but a normal quality of life. There are guys that play in the NBA after kidney transplant and an Olympic gold medalist in snowboard after liver transplant. There are folks in this room who have had a transplant, and the real victory beyond their presence is that you don't even know it. Transplant is, in fact, such a good solution for Americans with end-stage organ failure that it is a victim of its own success. The list is long. You'll wait for a kidney transplant for 5 or 10 years. Multiple solutions, attempts to increase organ donation rates, living donation, stem cells, 3D printed organs has, as of yet, failed to move the needle. Of late, there is hope that genetically engineered animals' organs may prove successful as a result of CRISPR technology. There is easy editing of animal genomes, and now pigs with 4 genes knocked out to make them less pig-like, and 6 human transgenes knocked in to make them more human, have had success in large animal preclinical models, much of that work done at Hopkins, and in several EIND applications. Now, United Therapeutics, Johns Hopkins, and NYU have received the go-ahead for an FDA-approved clinical trial to start next month, transplanting those genetically engineered xenokidneys into desperate patients. So these are exciting times indeed, and I'll thank Dr. McCary, Dr. Prasad, and the FDA for including us in this morning's conversation.
Thank you so much. Allison Barrett-Weiss.
I'd I'd like to start by thanking Dr. Prasad and Dr. Verdun for this invitation and to sit amongst some of the most esteemed people that I've ever been around. And to say that I'm humbled is an absolute understatement. My name is Allison Brent, and I'm a veterinary interventional radiologist and an internist by training. I spent my entire career developing medical devices for clinical unmet needs in patients. They happen to walk on 4 legs. Until 2015, when my second daughter, Quincy, at 5.5 months of age, was diagnosed with a rare, severe, non-degenerative, yet highly devastating neurodevelopmental neurogenetic disorder called Angelman syndrome. I was naive enough to believe that it would be easier to fix a single gene disorder than a feline ureter. So my reaction was to try to fix her. I was wrong. We've solved the feline ureter problem, but unfortunately, we've yet to solve the single gene disorder problem, though I feel strongly we are well on our way. I was honored to join the board of directors of a parent-founded and led advocacy organization called FAST, the Foundation for Angelman Syndrome Therapeutics, and become their chief science officer in 2016. I was tasked with leading the efforts to create a roadmap to get the incredible science the foundation and others had been funding out of a mouse and out of human cells and into humans. Supporting the infrastructure to ensure that the robust and rigorous science we were funding did not sit on a shelf if deemed appropriate, but would positively impact the lives of children like my daughter was imperative. Through the fundraising efforts of our tireless community, we raised the money to get 6 therapeutic platforms from mouse candidate to human candidate in under 24 months, including an ASO, an AAV gene therapy, a hematopoietic stem cell ex vivo autologous gene therapy, and a CRISPR gene editor non-viral delivered. The next steps were to get from human candidate to first in human, generally the responsibility of a biotech or pharmaceutical company. This is where everything changed. We created the science, supported the infrastructure, started a pre-competitive biomarker and outcome measure consortium, and had candidate therapies that industry was not advancing. The cost was too high, the indications too small, and the pace was too slow. Yet this affects potentially 500,000 people in the world. Not that small. So we had no choice but to take a venture philanthropy route and to do this ourselves. We could not let robust, repeatable, and transformative science that could change the lives of our children wait for someone else to decide if and when to advance it or watch it die in a publication. So we started our first company, Genetics Biotherapeutics, to advance an ASO. We could not do this on the backs of family fundraisers and needed angel investors. The program, with its positive phase 1/2 clinical data, was acquired by Ultragenyx Pharmaceuticals in 2022, which allowed FAST, our foundation, to further advance our AAV, our HSC, and our CRISPR gene editor through IND-enabling studies and into phase 1/2 clinical trials. As the environment for industry investment got worse, our scientific discoveries actually got way better. The need to create an innovative model to advance all opportunities for the 1 in 15,000 living with one of these diseases like Angelman syndrome became so very obvious. Golf tournaments, lemonade stands, and cupcake sales were not going to cover the astronomical costs of IND-enabling studies, manufacturing, and running a clinical trial, never mind the millions it takes to develop the endpoints that capture the symptoms of rare disease. This is when FAST, the foundation, launched an accelerator called AS2 Bio and really took the venture philanthropy model to advance all 3 of these programs, cell and gene therapy, which our AAV gene therapy just cleared its IND a month ago, and our HSC gene therapy and our CRISPR gene editor should be right behind in the next 18 to 24 months. I serve as the Chief Development Officer of AS2 Bio and its associated portfolio companies for each of these programs. I'm responsible for advancing the non-clinical aspects of the programs, leading the IND-enabling studies, and supporting clinical strategy and innovative trial design. Through my additional role as the co-director of the pre-competitive consortium, the Angelman Syndrome Biomarker and Outcome Measure Consortium, we are able to bring academics, industry, and clinicians together, as well as the patient advocacy groups in the field, to develop endpoints and biomarkers that can show both meaningful and measurable endpoints to families and patients, understanding and highlighting at every turn what actually matters to families, how this is highlighting in over 20 years of natural history data in this rare disease, what is the patient's and the family's lived experience? What does improvement look like to those most impacted by debilitating non-degenerative disorders that are equally debilitating to some of the worst cancers you can imagine? This is such a severe condition, and how can we best understand risk tolerance and patient and parent preferences? This is critically important as we think about drug development. So our story is only one example of how one community of thousands, the Angelman community, is on the verge of having opportunities to treat underlying neurogenetic conditions in one of the greatest ways possible, through genetic cell and gene therapy. And the pipeline is enormous based on the funding from families. We see the potential for an early win for other genetic neurodevelopmental disorders, Angelman is only one example. It is beyond debilitating and it's too risky for us to not take the chance to push this forward for our kids. So we ask for pragmatism in manufacturing, toxicology packages, and in clinical trial design. Controlled studies for these populations in cell and gene is not something families are accepting of. We have 20 years of prospective natural history data of the highest quality. Prior controlled studies that are in phase 3 should be required to share and support the future novel statistical modeling. We should not have to repeat controlled studies over and over again. In addition, we need to follow through and engage in outcome measures to ensure that we find the most meaningful changes with the patient at the center of what we focus, what matters to them. So today, I am here to wear whatever hat you want me to wear. The most important one being the parent, being a clinician, and being someone who understands what it takes to get treatments to patients that so desperately need them, like my daughter, Quincy. We are doing everything in our capabilities to develop therapies that are safe and effective, driven by only rigorous and robust science. And trying to do this with your support at the FDA is the most efficient way that we can make this sustainable for all impacted with such rare conditions. Angelman's only one of them. So thank you for bringing us all here.
Thank you so much. I really appreciate those comments. In the interest of time, just because we want to get through 8 people before the secretary joins us, I was given the suggestion that, and I know you all are appreciative to be here, and we're appreciative to have you, but we could skip the thank you portions and maybe skip the intro and get to the heart of your comments if you want to change your comments. Having said that, thank you so much, Brett Copeland.
Hi, thanks. I'll have no problem getting through our time. Most of my commentary was already discussed by those who can do it much more eloquently than I. So I run the Dystrophic Epidermolysis Bullosa Research Association of America. We're trying to cure and treat epidermolysis bullosa, one of 18 genes. I started my journey when my daughter, 17 years ago, was born with a particularly severe form of recessive dystrophic epidermolysis bullosa, which is the inability to produce collagen 7. It's a devastating disease. It's life-limiting. She's already had 2 squamous cell carcinomas, which means we are right now on a time clock. General population, 100% curable. In EB, 90% fatal within 5 years of first presentation. So we're on a clock. With that said, we're very lucky. We had our 2 years ago approved first redosable topically applied gene therapy. April 29th, CBER approved an autologous gene therapy approach. I want to applaud some of the things that Dr. Prasad, you have said recently. The accelerated approval pathway, fantastic, fully supportive, anything I can do can help. However, when insurance companies refuse to reimburse them, it doesn't matter if they get approved, right? So I would hope that perhaps you could write some guidance that supports the scientific rigor that went through even with the advanced approval pathway, talking about the safety and the efficacy that is there. You also referred to common sense, how decisions need to be common sense. Where I suggest is listening to the patient voice. The patient voice will determine some of those common sense solutions. Understanding the risk-benefit analysis, patients with rare diseases, specifically complex, life-limiting, will take more risk for an unknown benefit. They're willing to take on adverse events for an unknown, scale benefits. Incremental change, incremental improvement is everything to a person with a rare disease. I want to also talk about the harmonization between the FDA and the EMA when it comes to protocol design. We had a significant challenge. One of our cell therapy companies, the EMA had a more advantageous approach. The FDA decided to harmonize with that, and that allowed us to recruit the phase 3 trial in under 6 months. So the importance of the PRV, can't state it enough. Now they're running to try to get this approved by September 26, as fast as possible. They're looking to file their BLA. So it's incredibly important. With that, a couple last things, and then I'll stop. I will say with one caveat, my wife says there's never a microphone that I haven't liked. So I'm going to try to keep it quick. We've talked about the PPQ, trying to push things off as much into post-marketing commitments, which you guys are supportive of, potency testing. Most importantly, you guys are saying the right things, and I appreciate that. What we need to ensure is that it trickles down to the review teams. In some cases, it does not, unfortunately. So what I would suggest is a good thought, is as you onboard, or even now as they're there, to bring in a training module some patient advocates to discuss the diseases that they live with so they can understand the severity, what we live through. They can walk a mile on our shoes, because you will never understand, as Terry talked about before, the crushing nature, not only for the patient, but for the caretakers and for the siblings. And I'll make myself available, and I'm sure I can get Ron and Terry to jump right along, and Allison as well, I'm sure, to help recruit or be a part of those training sessions. And more than happy to be here whenever. So I'd like to thank you for having us here, and I cede my time.
Thank you so much for those excellent thoughts. Rachel McMinn.
Yes, I'm a trained PhD scientist, but I'm actually a sibling. My older brother suffers from a rare, debilitating neurological disease, and after I kind of went through a lot of soul searching through my professional career, I started asking myself, why are there no treatments for people like my brother? And I realized that there weren't any, because neuro is incredibly hard. I think we know more about outer space than we know about how many neurons are in the brain. So I decided that if I didn't do it, I didn't know who would. And that really led me to found Neurogene, which is a company focused on developing life-altering treatments for patients with rare, devastating neurological diseases. We work on a program called Rett syndrome, which is a debilitating disorder. These girls, have so many different manifestations, but one of the most heartbreaking is that they have no ability to communicate, either verbally or even non-verbally, so their hands don't work, so they can't do simple things like get a glass of water or tap for something that they want. So speaking for the families and as a sibling of not knowing what my brother thinks and is not able to care for himself, we developed an AAV treatment, and I want to, really give a shout out to Dr. Nicole Verdun and the entire team at the FDA that has been working with us. We are a START recipient, so we're part of the START pilot program. And talking about common sense, she's blown up through this program, just completely evaporated the alphabet soup of different type A, type B, type C type meetings, and we have a regular cadence of communication and interaction. It's been less than a year since we've had our kickoff meeting, and we're on the precipice, while we're collecting data from our phase 1/2, on the precipice of announcing a registrational trial design for our program. So no one in our history of our company, with all of our collective regulatory experience, has ever experienced anything like this to be able to get to this stage. We're also getting questions on what will be included in our BLA. So the design itself is meant to really generate and stand the test of time and be very rigorously driven, even though it's for a very heterogeneous disease. We're not looking at a placebo-controlled design, so I can rest assured that is not where the agency is at from our perspective. And it's been a breath of fresh air, to be honest with you. Very creative, but we're also getting questions about, helping us think about that BLA submission, so that when we get to that phase, we've already thought through it, because we've been constantly back and forth and interacting with the agency. So I want to say this is something that is going incredibly well, and I want to advocate for the learnings of START to be applied across the rest of CBER and, maybe even beyond that to other divisions of the FDA for these very high unmet medical needs. Beyond that, I can just say there's, obviously a lot of opportunities for efficiencies. You as the FDA, there's been over 6,000 patients treated globally with AAV. 6,000. So how can we use AI to mine that data to understand the kind of side effects and the monitoring that's going to be required to make gene therapy even safer? You have access to all these toxicology studies in your databases. You know, can you pool them and again use AI like you're talking about? How many wild type animals need to be injected with AAV yet again to you know, how does that answer the fundamental question of how it's going to behave in a patient? So just with that, I want to thank you very much for the opportunity to be here.
Thank you so much. Fyodor Orlov.
So I'll skip through the introduction, get straight to the core point. I was on the core team that gathered in early August 2024 to design, de-risk, and deliver the world's first CRISPR on demand to baby Kyle Jr. of Philadelphia. He's home with his family. And Dr. Prasad, I corresponded with his treating physician, Dr. Rebecca Arns-Nicholas, after I read your paper on parachute outcomes, and she agrees. Therapies such as what baby KJ just received fall precisely into that category, so thank you for that framing. So let me describe this could only happen in the United States origin story of this medicine, its path to baby Kyle, and what implications this has in terms of progress to make gene editing medicines available on demand for all American children who need them. So I work at the Innovative Genomics Institute at UC Berkeley. It was founded by Jennifer Doudna. She shared the Nobel Prize for inventing CRISPR gene editing. And the goal was to develop innovations to expand its public health impact. So proud to say we just did that, but critically we did not do it alone. So 2 years ago, we partnered with a leading American company, Danaher Corporation, subsidiaries of which are world-leading providers of clinical-grade CRISPR components. So in our partnership with Danaher, as in the case with baby KJ, we wanted to shorten the time and cost to CRISPR on demand from its current years and millions to months and just a few hundreds of thousands. Now, separately, we joined forces with 4 other teams in a program to develop accelerated CRISPR therapies. It was funded by the NIH. It's called the Somatic Cell Genome Editing Consortium. Shankar spoke about this. And one key goal of which is to share regulatory learnings. Okay, and so in early August 2024, all of these connections proved really timely. So Dr. Arns-Nicholas and her colleague at Penn Medicine, Dr. Kiran Musunuru, reached out to us with the news that there is a newborn at CHOP with a devastating genetic disease, urea cycle disorder, CPS1 deficiency. So we at the IGI immediately looped in Danaher and the race against time began. Okay, so what was the recipe for success? 2. Number 1, it was a collaboration between organizations with complementary, non-overlapping superpowers. We had CHOP, an elite tertiary care hospital with experience in caring with such infants. We had 2 major research universities, Penn and UC Berkeley, where the IGI is, putting to full use their deep experience in CRISPR innovation. But critically, a number of the tools we used had never been used clinically before, and some were provided by researchers at other nonprofits at universities, such as Shankar Tsai and David Liu. So now, the role of the private sector and industry was essential. Danaher companies, so IDT in Coralville, Iowa, Aldevron and Fargo, North Dakota, stepped in with innovation in manufacturing to make the clinical-grade gene editor in record time, because the baby's clock was ticking. Second key, the Super Office of Therapeutic Products of the CBER played an absolutely enabling and essential role. They provided regulatory oversight and guidance at every step, and most importantly, they prioritized the health of the baby. So Dr. Makary earlier said that, the FDA should challenge deeply held assumptions. I think they just did that. And I just really want to honor Dr. Nicole Verdun and her staff. They made baby KJ's return to his family possible. Look, I honestly think this can only happen in the US, where we have such clinical might in research innovation, industrial manufacturing power, and it's combined with a regulator, the FDA CBER OTP, that just welcomes such innovation. Last point. So in addition to my work at the IGI, I'm also a professor of molecular therapeutics at UC Berkeley. I teach how CRISPR works to hundreds of undergrads and dozens of grad students. Let me tell you, these incredibly motivated young people are on a clear path to becoming phenomenally talented gene editing medicine builders and gene editing savvy doctors. But I gotta say, we have to protect this talent pipeline. This isn't just about maintaining American biomedical leadership. It's basically about making sure that other nations, frankly, can't compete. Look, folks, we have an extraordinary opportunity to maintain American leadership. The story of baby KJ charts a fundamentally new path to genetic medicines on demand, scalably, and affordably. Our ultimate goal is to have the for-profit sector step in a major way and for there to be dozens of flourishing biotechs providing approved CRISPR on-demand therapies. But at the present time, as many speakers have mentioned, such companies are struggling under the old ways of making such therapies. The bright new path this four-way team has just charted needs continued support, as does the CBER OTP, to stay the course and get us to an entire generation of American babies who receive life-saving CRISPR on demand. Thank you.
Thank you so much. Jeff Allen.
Good morning. My name is Jeff Allen. I'm the president and CEO of Friends of Cancer Research, a policy and advocacy organization here in Washington, D.C. I have no personal disclosures, and my organization has received funding from 3 firms with marketed products in this area. As previously noted, CAR T therapy has emerged as a really key treatment modality for patients with cancer and possibly other diseases as well. For previously untreatable cancers, major improvements in patient responses, including complete responses, are occurring alongside significant improvements in progression-free survival and extension of overall survival. In some cases, for some patients, this is over 10 years after their initial treatment. However, the progress in CAR T therapy has been predominantly for the treatment of hematologic malignancies, but recent data is showing that this could change, and if we can get the correct policies in place, this will be far more commonplace. For example, this past weekend at a leading cancer conference, we saw the promise of CAR T therapy for previously hard-to-treat conditions like glioblastoma. This is one of the most common and deadly forms of adult brain cancer, and the research group leading the study at the University of Pennsylvania saw tumor size reduction in over half of the patients treated with the new dual-targeted CAR T therapy. And this really signals the potential hope for CAR T therapy to be able to address unmet medical needs in currently hard-to-treat cancers. So as we think about ways to further optimize the development and regulation of cell therapies, for a few future opportunities to highlight. Number one, the rapid identification of candidates for early phase trials. Due to current limitations in preclinical models and limitations in evaluating the vast number of potential constructs, novel approaches for efficient screening, and prioritization of gene editing candidates, new ways are needed to ensure the most promising therapies advance into clinical development. Number two, new pathways to facilitate access. Particularly for rare cancers and other small populations, uncertainties in regulatory flexibilities and manufacturing requirements can create hurdles to sustainable development, often limiting commercial interest. A new structured approach is needed that balances the regulatory oversight with operational feasibility alongside patient need. And third, the use of real-world evidence. Leveraging data from current clinical practice can be an important tool for longitudinal monitoring to verify outcomes. A framework for prioritizing, collecting, and analyzing real-world data could help support timely approvals to address unmet needs and support long-term benefit assessments. We certainly, as a community, look forward to working directly with the FDA and appreciate their support thus far and bringing together this wide group of stakeholders for this important discussion. So I think as we think about new opportunities for efficient translation of new discoveries that ensure patient safeguards and maximize the potential for cell and gene therapies to successfully address the needs for patients with limited treatment options. Thank you.
Thank you so much. I see the NIH director has already arrived, and we're just waiting for 2 more people, and we'll take a little break, but then I will ensure that everyone gets a chance to speak and that some of you may speak in their presence. I'll turn to, next on Tom Cahill.
Great. It's great to see so many familiar faces. I'm Dr. Tom Cahill, founder of New Path Partners in Boston, Mass. Let me begin with what matters most. Children once expected to live in wheelchairs are now walking. Teens with sickle cell disease are going years without a single crisis, and individuals born blind are seeing for the first time. These aren't projections, these are real patients benefiting from American-made cell and gene therapies, developed in US labs, funded by US taxpayers, approved through US regulatory leadership. But that leadership is eroding. Over the past 3 years, more than $150 billion in biotech market value has disappeared. Nearly 70% of biotech companies are either trading below enterprise value or valued under $100 million. More than 60% have less than 1 year of cash remaining. IPO markets are frozen, private capital is retreating. This isn't a failure of science, it's a failure of confidence, driven by regulatory opacity and unpredictability and exploited by a foreign adversary eager to fill the void. While we hesitate, China is accelerating. Chinese companies now bring drugs to market in just 4 years, less than half the US timeline. Their clinical trials cost up to 90% less. They reverse engineer US discoveries with state-backed chemists, file sweeping global patents, and already account for more than 35% of newly licensed drugs, up from less than 5% just 2 years ago. In 2024 alone, more than $40 billion flowed into Chinese biotech. From where? Most of it from American venture capital and big pharma. We banned Chinese chips from our missiles and blocked their cars from our roads, yet we still allow China to steal taxpayer-funded medical breakthroughs, conceal clinical data, and sell these inventions back to us, often as medicines we put into our children's bodies. This isn't just an economic risk, it's a national security threat. If we stay passive, China won't just dominate the future of medicine, they will control the infrastructure, the data, the supply chains, and the standards. That's a chokehold on American health, sovereignty, and innovation. But it's fixable. The president has the power to act. Congress can step up in what should be a rare and obvious bipartisan priority. HHS must raise the alarm, and the FDA already has tools. We simply need to use them. The FDA also needs to evolve how it thinks. We need a cost-benefit mindset, a Bayesian approach. Not all diseases carry the same risk. Not all therapies require the same level of evidence. Treating every indication with a one-size-fits-all model leads to unnecessary delays, ballooning costs, and lost lives. A rare pediatric disease with no treatment available should not be held to the same trial standards as a low-risk, well-served indication. The system must be calibrated to reflect urgency, severity, unmet need. We should align early development requirements with true clinical risk, allow IND submissions before final GMP release, publish metadata from IND packages to restore public trust and improve clarity. Incentivize bold, high-impact science with fast-track pathways, review vouchers, and guaranteed market support. This isn't deregulation, it's discipline modernization. It's about building a system that's fast enough for today's science without compromising safety. Because without action, American lives will be lost, not to science, but to delay. Biotech isn't just about breakthrough cures, it's about national strength. We created the industry, we can still lead it, but we need to act. Thank you.
Thank you so much. Bambi Grilly.
Hello, I run the Regulatory Affairs Program at the Center for Cell and Gene Therapy in Baylor, and I'm also the Chief Regulatory Officer for ISCT. The Center for Cell and Gene Therapy has filed over 100 first-in-human INDs and 150 clinical trials. From that, due to the cost of development, a number of startup companies have been initiated, and I've served as a consultant for 8 of those. Almost 50% of those companies have failed due to cost issues, and the other 50% are overseas, which I emphasize just to say many of the points that other people in this room have brought up are real concerns that we need to talk about when we talk about drug development or cell and gene therapy product development. I have so many agreements with a lot of the things that were already said. There's 2 things I want to add to that. One is that I think that we really need to consider formalizing a gradient of GMP for early phase studies. That has to be something that enters the conversation. And we also need to have a functional cost recovery system for drug development that's happening in academic medical centers. And to keep this short, I just want to close by saying that one of the clients I work for, and also at ISCT and the Global Regulatory Summit, a conversation was had about how can we run clinical trials overseas and then bring the products back so that we can get approval in the FDA. That has to be a conversation that we want to get away from. We need to move back to being able to successfully conduct these studies here and get to market. Thank you.
And that point's well taken. Sean Morrison.
I direct Children's Medical Center Research Institute at the University of Texas Southwestern Medical Center. I'm an investigator at the Howard Hughes Medical Institute. I believe deeply in the FDA mission. I've been an advisor on the Cell, Tissue, and Gene Therapies Advisory Committee for several years. I'm a co-founder of a company called Stratus Therapeutics, an advisor to Inception Therapeutics and Conception Biosciences. But I don't want to talk about any of that stuff today. I'm here as a representative of the International Society for Stem Cell Research, which has done a lot over the years to promote the clinical translation of stem cell therapies and worked a lot with the FDA. We've appreciated the opportunity to partner with the FDA and look forward to that opportunity going forward. There's a number of initiatives at ISSCR that are designed to focus on priorities that the FDA has stated, like the development of non-animal models for disease and accelerating the development of therapeutics. But I just want to, I want to focus on a part of this issue that hasn't been addressed so far. You've had really outstanding ideas this morning from serious people with deep insights and clinical evidence of safety and efficacy, but there are also bad actors in this space who are attempting to sell essentially snake oil to rip off desperate patients by capitalizing on the promise that you hear from the research in this room, but who are instead selling snake oil. These companies never do clinical trials. They have no plans to do controlled clinical trials. They're operating illegally, but it's estimated that there's 2,000 companies like this in the United States. They commonly claim to be able to cure diverse medical conditions with one-size-fits-all therapies, even conditions for which there is no plausible scientific rationale or stem cell therapy. For example, there's no scientific rationale for how umbilical cord blood cells or placenta cells could be used for the treatment of Parkinson's disease or Alzheimer's disease or orthopedic conditions or neuropathies, yet there's hundreds of companies out there that are selling to Americans making that claim. Americans really need the FDA to protect them from those people. Buyer beware is not an effective strategy when it comes to experimental medical therapies, because even most physicians don't have the expertise to evaluate which experimental therapies really have merit and which ones don't. Companies, what we've learned is that companies that ignore FDA regulations also commonly ignore good manufacturing processes. Scores of people have become septic as a result of injection of these products. And so I'll just end by saying that the challenge here is to find ways of accelerating the development of real therapies with sound scientific rationales, clinical evidence of safety and efficacy, without deregulating to the point where you open the doors for the bad actors to sell snake oil to Americans. It would undermine confidence in the agency, and it would undermine confidence in the field in general.
Thank you so much for that important point. Michelle Rangarajan.
Thank you so much. I'm a physician scientist at Mass General Hospital and affiliate faculty at the Broad Institute, but I'm speaking here in my personal capacity and don't represent my institutions. I'm scientifically trained as a cell biologist and systems immunologist, and now run a lab focused on Duchenne muscular dystrophy and genetic medicine, particularly as these intersect with tissue immunity. I also started and run a very new academic industry advocacy consortium that's focused on humoral immunity to AAV, with a particular emphasis on seropositive individuals. These are the substantial fraction of patients who have prior environmental exposure to AAV viruses that deliver gene therapy, and so are excluded from nearly all gene therapy trials due to concerns about both safety and efficacy. Most importantly, in driving all of this work, I'm also a mom of 2 extraordinary preschoolers who live with Duchenne muscular dystrophy. As the last speaker, I want to echo and underscore that we are at an incredibly exciting moment for gene therapy and genetic medicine. We are so remarkably close to achieving escape velocity for Duchenne and so many other inexorable genetic diseases, where patient-focused scientists, clinicians, and regulators can enable the safe and transformative therapies that could completely change the trajectory of life for patients. But nearing escape velocity also means that we need to continue and accelerate the pace of therapeutic development so that people alive today can access the exciting science and clinical developments that we are talking about. I want to underscore that there is substantial need in the pediatric rare disease community. We've heard that most rare diseases don't have an FDA-approved therapy, but even in diseases like Duchenne, where an approved therapy exists, many, many patients are still left behind. By some estimates, well over half of individuals with Duchenne don't have access to gene therapy, whether because of seropositivity, higher risk mutations, or underlying cardiac risk and frailty. Splice-switching oligonucleotides are showing very exciting developments, and these may soon be available for 30 to 40% of patients with this disease. With platform designations and umbrella trials, another 40% of patients could access these treatments, but they can't now because, like chronic granulomatous disease and like SPG50, each of them individually have mutations that are too rare for commercial interest. At the same time, we cannot let a quest for cures be the enemy of disease mitigation. Incremental advances not only provide meaningful quality of life, they also set up patients to be healthy enough to access future better therapies. In 2025, a 20-year-old man in a wheelchair who can lift his arms, type on a computer, and put on a headset has a fundamentally different ability to interact independently with society than someone who can't do those things. A therapeutic product that preserves that ability for a few more years would be a massive win. Finally, I want to touch on two practical challenges. As a clinical endocrinologist, I recognize that one way to accelerate therapeutic development, really change the entire field, is to enable the use of surrogate markers. I'm grateful that the Commissioner and Dr. Prasad recognize the scientific and clinical challenges of running trials in rare disease, where small populations with superimposed heterogeneity can make it very easy to miss efficacy within the timescale of a clinical trial, and where terminal diseases raise real ethical concerns about long placebo arms. I appreciate the recognition of the importance of accelerated approvals with strong post-marketing commitments for drugs with biologically plausible mechanisms and strong genetic basis in rare disease. However, the qualification process for surrogate biomarkers can itself be quite challenging and inefficient. I certainly would appreciate greater clarity on how such markers combined with real-world evidence can be better optimized, particularly for diseases where the risks of not treating are very clear and remarkably high. Lastly, as others have mentioned, I worry that the natural siloing of information by disease or clinical specialty or within a scientific community can make it difficult to identify and appreciate class effects that, if not appropriately managed, can lead to adverse events and death. I know that many people in this room are committed to and working hard on data sharing, and I look forward to working more closely with regulators to advance these kinds of sharing of learning and knowledge. I'll just end as the last speaker by echoing Dr. Hunt and saying that for patients and their families, the single most important commodity is time. And as you think about improving efficiency and advancing therapeutic products, I hope that you continue to remember that for many patients, time is muscle, time is brain, and as Dr. Hunt so clearly said, time is life. Thank you.
Thank you so much for that. And thank you for all the panelists for coming here this morning and sharing your perspectives. I made a list of some of the themes I heard. I'm going to read a little bit of this list. Many of these things are on the radar of the Commissioner and myself. Some of these things are not on our radar, so I'm glad you put it on our radar. Some of the things I heard is what's the role of the FDA and what's the role of local IRBs, and can we perhaps negotiate something so that it's not China that's going to be launching these studies, it's going to be the United States. Pediatric and rare diseases are completely neglected and things completely on our radar. You made an excellent point, Sean Morrison, about compliance. We have a compliance office that, you're absolutely right, we want to allow the innovative companies to thrive, but we have to regulate the bad actors. We can't let that taint what we're doing here at FDA. I've heard China mentioned many times. You all know it is a priority of the Commissioner and this administration that we need to retain control over this important intellectual property. Flexibility is a theme that came up repeatedly. We are fully committed to being flexible. Diseases that are rare, dire, or unique constellation or patterns cannot have the same paradigm as high blood pressure and high cholesterol. I've heard about control arms. You're absolutely right. What should the control arm be in a study, and which studies will not even need a control arm? This is something we need to talk about. Many speakers mentioned AAV vector delivery and the risk of immunogenicity and things of that nature. We also have now lipid nanoparticles that avoid some of these pitfalls. So we are making biological advances as well, and that's an important point I wanted to bring to everyone's attention. Finally, some people asked about learning from other experience. Can we learn from these things? I will tell you one of the Commissioner's priorities is what documents can we take at FDA, redact, and make available publicly so that you all can leverage it, apply AI to it, and learn from this experience. We have a treasure trove of data here at the FDA. It is not being optimized and utilized, and it is a priority of this administration for issues of data sharing and transparency as well as learning. So, having said all those things, those are just some of the themes I heard and jotted down here, but I heard a lot of very specific, very interesting idea about who should counsel people coming in as reviewers, et cetera. So many interesting ideas that were not on our radar. We really appreciate this. This is part of a continuing conversation we're gonna be having with you. We're gonna continue to learn and try to incorporate these ideas you're giving us. Now, I'm being told that we have the Secretary of Health and Human Services, Robert F. Kennedy, is here. We have Mehmet Oz from CMS, and I saw Jay Bhattacharya already from NIH. We're going to take a brief 10-minute break. We're going to regroup, and we're going to hear from those speakers, and then we're going to allow you all to have a conversation in their presence to directly put your concerns, not just to FDA, but to the broader HHS, which has abilities that we don't fully have here. So thank you all for doing this. I really appreciate you coming out this morning. Thank you.
Okay, we're just going to start. If it's on video and live stream, great. If not, we'll just go. So thank you, everybody, for your incredible contributions. We hear you loud and clear. I got a good dose of the first half, and the second half heard remotely while I took some meetings. Thank you. And there were many common themes. We're going to take those messages very seriously. This is not a horse and pony show to say we did this. This is an honest listening session, and we're going to keep going. We're not going to stop listening. This afternoon, we have about 100 pharmaceutical CEOs and heads of R&D coming to the FDA, and we're going to be doing a town hall listening forum with them. We want their ideas, and we are going to be an FDA that listens. We're very honored to be joined by the Secretary of Health and Human Services, Robert F. Kennedy, by the Administrator of CMS, Mehmet Oz, by the Director of the NIH, Jay Bhattacharya. So thank you, gentlemen, for joining. I just want to start off by saying this has been an incredible conversation on cell and gene therapy, one of the most cutting-edge areas of all of clinical medicine. And I think we can learn a lot from individual experiences. We tend to talk in the lexicon of clinical medicine about level 1, 2, and 3 evidence, and it's become this sort of dogma that we can only do things with evidence, and the evidence is categorized in this artificial framework of level 1, 2, and 3. 1 is defined as a randomized controlled trial. Well, that is true. Level 1 is better than 2, and 2 is better than 3. And we want to do things where evidence exists, and we want to pursue the creation of new evidence. But 60% of clinical decisions are purely discretionary where no evidence exists. And we can learn from individual cases. We can learn from individual cases. Some of my former colleagues described a case of stage 4 glioblastoma multiforme, generally considered to be incurable, as you know. And there was a case of a long-term survivor. Now, when somebody survives long-term, sometimes we think they were misdiagnosed, the pathology was read incorrectly. But this original pathology diagnosis was re-reviewed every year in a clinical conference, and it was stage 4 GBM. Why was that individual a long-term survivor? Can we ask these big questions that don't fit the model of an RCT? It turns out, it's interesting, that patient had a resection of the tumor and then an infection of the resection bed, and the abscess in the brain led the surgeons at the time to do the standard of care, which was to remove the skull plate, drain the infection surgically, and then remove in a second stage procedure down the road the skull plate. What happened? Maybe something happened. Maybe the bacteria activated an immune response. Maybe the bacteria had some endotoxin that chewed away at the tentacles of the remaining tumor in the resection bed. We don't know, but we can learn from individuals. And you saw that we took action immediately on banning 9 petroleum-based food dyes from the U.S. food supply. Well, we can, yes, read the randomized controlled trial on petroleum-based food dyes in Lancet, but we can also listen to individual families who say that they have had a kid with aggressive behavior, and when they stopped all the petroleum-based food dyes, single elimination of just food dyes, the kid's behavior then became normal. And then when they regressed, it was because they were reintroduced a year down the road. When you have a story like that, when you have an adverse event that is ignited seconds after the administration of something new in a young, healthy individual, that's causal until proven otherwise. We can learn from individuals. So I think that is the approach we need to take with cell and gene therapy. With that, I want to say perhaps Dr. Bhattacharya, you can start off, and then Dr. Oz, and then Secretary Kennedy will close us off.
Thanks, Dr. McCary. And it's an absolute delight to be here, especially since, for me as the director of the NIH, I get to brag about some of the advances that have come as a result of NIH investments over the past years. But I'll start further back than that. When I was a medical student, I learned about sickle cell anemia. And those of you who know about it, almost all, I'm sure all of you in the room know that it is caused by a single gene mutation in one protein that causes a whole cascade of problems resulting in painful episodes, early death, a huge problem. And I remember reading about it, just being first amazed by the elegant biology and the understanding of it that I learned in medical school, but also shocked and just saddened that there was no way to treat it. I never imagined in my lifetime that we would actually have the capacity to actually cure it. And it is cell-based therapy, gene editing that has allowed us to cure it. And actually, it's not even the knowledge of the mutation that causes the misfolding that led to it. It was a whole separate line of biological knowledge about how our body switches from fetal red cell production to adult red cell production. And then the knowledge of how to change that switch in an individual. That's what's led to the cure. It's an absolutely amazing tour de force of biological knowledge applied in clinical settings and then advanced so that we can actually deploy it at scale for patients at large. Patients with sickle cell anemia now have real hope that they will be able to live long, productive, healthy, happy lives. And that was unimaginable to me in medical school. I'm old, so 30 years ago. So that's the promise. The challenge, though, and you all face this every day in your work, is how do we make those advances scalable and affordable? It's expensive to do this, and it's more expensive to let a patient have sickle cell anemia and die, but that's much more expensive, so it's worth it. But the question is, how can we use this knowledge and scale it so that it's available to everybody with sickle cell disease, not just the select few that happen to have access to this care? We would love to be able to scale it so that the whole world could have access to it in an inexpensive way, because sickle cell isn't just happening in this country. And so that's going to take more biological advances, but it's also going to take a deep understanding of how you translate those biological advances into actual therapies that treat and manage the conditions that people have. And it's also going to take, on top of that, a regulatory framework that makes sure that the things that do advance for patient care are the ones that actually are safe, that have excellent evidence behind them. The other thing I was looking up just before I came here was this amazing case of this little baby KJ. I actually got to brag about it a little bit just because it was an NIH-funded work that actually led to this, where this baby with a very rare genetic disorder, affecting I think 1 in a million kids, the gene editing technology using CRISPR as its key tool, where this kid would normally have a 50% chance of living to age 1, his problems are addressed in a way that actually was unimaginable just a few weeks ago. So what we have is a tremendous promise, but it's a 1 in a million condition. Just as Dr. McCary was saying, could you run a large randomized trial on a condition that's 1 in a million? No. So you have to have different standards of evidence for these wonders. But we still have to have excellent standards of evidence. We have to assure that the therapies that the patients are getting are actually going to work for them and then are scalable. And I think that's the challenge I pose to you all and also to myself and to the scientific community. How can we assess these technologies so that the wonders that they're there can be sifted away from the things that look promising but are actually real? And then how can we scale this so that it's affordable to everybody? Thank you.
Jay, thank you very much. Cell and gene therapy. We can talk about it in many contexts, but I think from the perspective of CMS, Medicare, Medicaid, its primary difference is we're talking not about treating of symptoms or chronic illnesses which can be ameliorated, but actually true root cause solutions that in some cases could result in permanent cures, even in kids and adults sometimes who had illnesses we thought heretofore untreatable. And so it changes dramatically the dynamic, completely within what Maha would have hoped in his wildest dreams, which is actually get at the main reason you have the problem and eliminate it so you no longer have to deal with those issues. But to do that, you often have to make significant upfront investments, which this FDA has been brave enough to tackle, and this NIH has been wise enough to research. And we believe if we can continue this trend and CMS can pay for these solutions in ways that make sense for many of the folks in this room who are involved in industry, and for the researchers, allow your ideas to reach the markets with more likelihood, we'll be able to put this, the entire process on jet fuel. So let's talk about that. First off, how do you provide access to transformational therapies, ones you know work but are really expensive sometimes to develop and distribute? How do you do that in a way that might actually plausibly offer savings, value to the American people? And the savings aren't always monetary. You save a life, that's worth a lot. But how do you make the system work in order to reward people appropriately for the investments of time and money that they've made? And what are the real-world efficacy realities? How do you get the manufacturers accountable to the process so they're also engaged, they've got skin in the game? I don't think you can do that if you don't collaborate. The reason this meeting is so important is because it will allow us to put on the table the real issues that hinder our distribution of life-saving, life-changing modalities in an effective way. So we all have probably at this point friends who have been on CAR-T and have their lives saved. We've had friends who've got stem cell therapy, even if it's for joints, sometimes much more serious problems than arthritis, but they can be meaningful and life-preserving as well. Folks who've regained vision with some of these therapies, CRISPR has become a household term. These are changes in how we even think about the ailments that we had given up as untreatable. But there's one illness I want to talk about in particular because we've been involved in it already, and that's sickle cell anemia. Abe Sutton, if Abe can stand, Abe runs CMMI. I brought the whole team here. Abe is a flight risk, but I think he's in the room. There he's in the back. But CMMI is, well, he's standing to the left. If you've got questions about the CMMI models, go talk to Abe because he's brilliant at it. But in sickle cell was an interesting predicament. You've got a treatment that costs, we believe, about $2 million, but you potentially cure a kid with sickle cell anemia. So how do you do that? Who's going to pay for that? Especially when insurance companies know patients stay there for 18 months, sometimes shorter. So you do it because you get smart. First of all, kids are at stake, so that actually increases the opportunity. But you got to ask the right questions. And I think that's what this session in part should reveal. What are the right questions to ask? Because without them, we'll never get the right answers. And we need advice. We don't have all the answers. What Abe was able to do with his colleagues at CMI, and this started before he came in, was to begin looking seriously at what are different ways that you might be able to pay for some of these services. Do you create a reinsurance platform so that we socialize some of these costs? Because we know that the revenue is there. The value to the system exists because if these kids aren't continually hospitalized and in chronic pain with complications that are incredibly expensive and don't provide value oftentimes, we're going to save the system a lot of money. If we provide outcomes-based reimbursement and solutions, that's a smart way of thinking about it. If your solution works really well, you appropriately should be investing in that idea, and we should be compensating you for that. What Abe was able to do was to create tools that would allow us to focus on discounts when you bring the product to market so that people can afford to deliver them, and then payments over several years based on outcomes and deliverables that were mutually agreed upon. All of these make sense, but they only make sense if we're brave enough, if we're wise enough to make the investment of time and put ourselves out there. And to take these risks requires courage. And I think that's in many ways what the MAHA movement has been about. And the person sitting to my left, Secretary Kennedy, embodies that more than any other person on the planet because at its very core, he's a curious person. And curiosity means you're willing to ask uncomfortable questions, like how do you pay $2 million to help a kid with sickle cell anemia? But if you ask that question, which Secretary Kennedy has done, and you're brave enough and courageous enough to take the answers that are uncomfortable and share them, you'll achieve what CMI has done, which Abe has gotten 84% of African-American kids, primarily who are eligible for sickle cell, to be in states who now are part of this platform. And by doing all of the process together, by going out and purchasing and working deals out with the two main manufacturers and allowing those medications to be accessible and now affordable for these children, you are now looking at treatments that are available, again, to 84% of the kids in the country, and eventually we hope all. But it doesn't happen without strong leadership from the top. And the reason that Marty J. and I are here and will come in every time we're called upon is because of Secretary Kennedy. Thank you.
Thank you. Thank you, Mehmet. Thank you. Well, thank you, Mehmet, and thank you, Marty. A couple days ago, somebody asked me what my administration philosophy was when I approached, and I said it was to surround myself with people who are a lot smarter than I am. And, I have this extraordinary array of just of mental, intellectual power here with Mehmed, with Marty McCary, Jay Bhattacharya, and Vinay Prasad. And, but more importantly, we're friends. We talk to each other every day. We go to dinner with each other. We go on vacations with each other. And we genuinely enjoy each other's company. All of them share the virtues that I think that Mehmet just mentioned about curiosity, critical thinking, a willingness and an inclination to challenge orthodoxies, which is so important if we're going to have advances in medicine. Every advance in science and medicine throughout history has come from people who were challenging consensus. Everybody who is running this agency right now, all of these people were censored during the COVID pandemic. They're all renegades, and they're renegades because, one, they have courage, and most importantly, they have curiosity. My uncle, President Kennedy, the first time he met Jackie Kennedy, who would later become his wife, she was a reporter, and she asked him what he considered his greatest virtue. And he surprised her because she thought he would say courage, because he'd won the Pulitzer Prize for writing a book about courage, Profiles in Courage. He'd been a war hero in World War II, and the only American president who won the Purple Heart and a lot of other awards. But he didn't say that. He said curiosity. And that, I understand what that means. It means that he's interested in other people, and he has a mind that won't accept received truths, but is constantly asking these important questions. And I think that's what we need to do as an agency if we're going to meet our ambition that all of us share, which is that we transform this terribly wasteful system that we've inherited, that is now costing $4.5, almost $5 trillion a year total health care. The government side of that is about $1.8 trillion, that our agency manages. It makes our agency the biggest agency of the federal government. We are double the size of the Pentagon, and we are the fastest growing. We're growing, the healthcare costs in this country are growing at 2% every year, faster than the economy. So it's unsustainable. But more importantly, we have a sick population. We have, when my uncle was president, I was a 10-year-old boy. 3% of Americans had chronic disease. Today, 60% do. And it's debilitating our country. It's an existential threat, not only because the economic cost, 75% of American kids can't qualify for military service. We have a fertility crisis where American teenage boys have half the sperm count, half the testosterone as 65-year-old men. Our girls are reaching puberty 6 years early. We have diabetes and obesity crises that are rivaling now the Pentagon budget. So, and we can't continue. We need to transform ourselves to a healthcare system rather than a sick care system. And in order to do that, we need to unravel all these perverse incentives that really this whole agency is a bundle, our whole healthcare system is a bundle of perverse incentives where people make money by doing bad things. We're not at every level, the doctors, the other providers, the hospitals, the pharmaceutical companies, the insurance companies all make money by keeping us sick. And we need to fundamentally change that so that we are focusing on curing disease and making people better. And the way that we're going to do that is by unraveling those incentives and really focusing on curing disease. And you are so important to that because that's what you do. You're not offering us lifetime treatments. Mehmet often points out that, when my uncle was president, we spent 0 on chronic disease in this country. We now spend $1.7 trillion a year. The first chronic disease, the first continual treatment was the birth control pill. Nobody was taking pills every day. Now 60 or 70% of our population is taking pills every day just to live, just to get up in the morning, to have energy, to be able to be barely functional. And we need to change that. And we're going to change that through changing the incentives, but then more importantly, unleashing innovation. And particularly innovation that of the kind that you are particularly interested in, which is curing chronic disease. And I've seen the miracles that you can produce. This morning, I spent 20 minutes on the phone with Kyle and Nicole Muldoon, whose little boy, who's now 10 months old, KJ, got one of these treatments for his liver dysfunction. His liver can't process proteins. And through this CRISPR technology, I spent that time also, his physician, Rebecca Ahern Nichols, who's from CHOP, was on the phone as well, and she's an NIH-funded researcher. And they've developed something that has allowed this child now to process protein in his liver. And when I was talking to them, the child was sitting KJ was sitting on a chair playing with his sister. And that's something this child had a 50% chance of dying in his first week of life. So we can give the gift of life to people. We can give the gift of good health to people. And I was in April, I was in South Carolina. Mehmet was talking about sickle cell. I was in South Carolina with inking the final documents on a deal that we've put together with a number of other people, but the insurance companies, the drug companies, the local hospital systems have put together the deal to make this particular cell and gene therapy, available to every person in the state who has sickle cell, with being fully paid for. And that was a model that we've created, and we're now rolling that out. We're negotiating now with 38 other states. By the end of next year, we hope to have this available to every American. And it's saving the insurance companies money. We have to talk them into this because Mehmet points out it's the way that they look at their business plan forces them to look at short-term profits. But if they look at the total costs over the lifetime of that child, it's an enormous savings, even though it's a very expensive drug, but it's a one-time treatment. And more importantly, I spent the day with a young woman who really was in agony, a kind of hell her entire life because of sickle cell, of physical pain, of just terrible disability, of complete dysfunction. And she now has a full life because she received this treatment. So if we can give that to people, that is almost providential power that you have and that we want to unleash. And we want to make sure that we keep this country competitive with all other nations, that we're the hub of this kind of technology around the world. We want to make sure that we are getting rid of the regulatory impediments that stop you from getting your drugs to market. We are going to continue to figure out new ways with Marty of accelerating approvals for rare disease, for drugs and treatments that treat rare diseases. And we're going to make this country the hub of biotechnology innovation. we want to do that. One of the things I would ask you is that President Trump has given us an executive order that says for every regulation that we put in place, we have to get rid of 10. So I would solicit right now a list from all of you of any regulations you think we ought to be getting rid of, because we need to get rid of a lot of them in order to do the things that we want to do. If you can provide us with that list, and we will look at it and do everything we can to get rid of the bad ones and to put in place the good ones and then get you the funding. There's a lot of people who say, who criticize us because they say, well, we cut the budget of the agency. We didn't cut any of the budget of the agency. We have that money, and we are taking it away from the administrative costs and redundancies, and we're going to reapply it to spawn and foster and fortify innovation and unleash it. We consider you our partners, our allies. We're going to rely on you to come up with the solutions, and we're going to do everything in our power to sweep away the barriers from you getting those solutions to market and getting them funded, and do everything that we can to support you. Thank you all for being here today.
Thank you, Mr. Secretary, and thank you for believing in us and telling us to go and run as fast and as far as we can wherever our scientific discovery takes us. And so I can't thank you enough for that, for believing in us and letting us pursue these deep scientific endeavors. Thank you, Dr. Oz, for believing in cell and gene therapy and thinking about creative ways to pay for it. And thank you, Dr. Bhattacharya, for also being here. It's a testament, I think. You'll hear the different agencies have infighting in the past, and I'm sure somebody will suggest it in the future, but you can see that we are a team. We work together. We talk about doing things on the phone daily, fast, coordinated. And so this is one of the commitments we have, is to cell and gene therapy. We are learning, we are listening. We have to challenge some deeply held assumptions. Why does it take 10 years for a drug to come to market? Why are we spending over $4 trillion on healthcare and yet have to tell a kid with a rare disease we can't afford a treatment? Why do so many of our research studies fund treatments and not look at root causes? We have to challenge deeply held assumptions. So I hope that all of you will work with us as a team to figure out these big questions. And with that, I'll turn it back over to Vinay Prasad for the remaining 10 or 15 minutes. And thank you, Vinay, for leading this panel today.
Thank you, Dr. McCary. So first of all, my tremendous thanks to the panelists for coming and sharing their perspectives, and also what I think is truly unprecedented. It's rare that you have the NIH director, the FDA commissioner, the HHS secretary, and the CMS director all in the same place to listen to your concerns. I'm going to summarize real quick some of the top points that were brought up in this discussion so that you all hear it, and then I'll try to toss it to a couple of the panelists to directly advocate to you on the issues that matter to them. But some of the themes that we talked about that I think will be of interest to all of you, one is that cell and gene therapy is important to vitalize in the United States, lest we cede that dominance to China. And that's something that several of the panelists brought, and something of extreme concern. One of the panelists pointed out that it takes 10 years, Dr. McCary pointed out it takes 10 years in this country. China apparently is trying to get that down to four-year pipeline to get a drug approved. And I think that none of us here want to see U.S. dominance lost. Organ donation was a topic that was brought up, Secretary, and we are working at FDA on xenotransplants, taking genetically engineered organs from pigs and other animals and transplanting into humans. But I know there are a number of initiatives at HHS that can be done to encourage human transplantation. We learned a lot about regulatory flexibility, control arms, IRBs, and other requirements. That's within FDA. We'll tackle that. But cost and innovation and incentive, Dr. Oz. This was something that was brought up. And a number of the people here talked about how for a small disease population, it takes so many years and so much venture capital funding to even develop these products. Is there a way or role for CMS or other, or NIH to kind of support that initial investment, knowing that on the back end, we may get great returns? One of the issues that was brought up particularly was pediatric and rare disease. So I think I'll turn to a couple of the panelists to directly advocate for that issue. I think I'll start with Catherine Bullard and then Crystal McCall.
Thanks so much, and I'm Kath Bollard. So I am a pediatric bone marrow transplant physician. I sort of grew up, if you like, in the cell and gene therapy space. I always knew that it had promised to cure children with cancer and other chronic illnesses at a time when no one else really believed in the how this delivers. The talking point I really want to make is for children with solid tumors who relapse from conventional therapy, we are still giving them the same chemotherapy that we were giving them 40 years ago. There have been no major approvals for pediatric solid tumors. And as you'll hear from Dr. Mackel, her group and ours and many other groups here and nationally are developing curative cell and gene therapies for these children who have lost all other hope for survival. And this is a real gap because Big Pharma don't see the business model because it's a rare disease. And on that note, can I defer to my colleague, Dr. Mackel, to follow on for solutions?
Oh, thank you, Kath. And, you know, I've been trying to cure children's cancers using immunotherapies for 40 years. And, you know, the war on cancer that President Nixon launched in 1972, it did pay off. We are seeing now in 2024, we have amazing tools to cure cancer, and children are no exception. Cell therapy is able to control and regress tumors that no other therapeutic can touch in children's cancer. So the science has delivered. But what's wrong? We can't get these things to market. There's a problem with the finances, the economics. Business as usual, the biopharma model will not work. We need new models. We need public-private partnerships. We need FDA and regulatory flexibility so we can reduce the cost of development. We need to get CMS and the private payers on our side. 50% of children in America receive their health care coverage through Medicaid. Any kind of approval, we're so delighted to hear Commissioner McCreary's idea about a fast track to approval for rare disease. But if it is in any way reduced from full approval, we'll end up with an approval and we still won't be able to get the therapy paid for. So it really is going to require working throughout government at every aspect, all of the folks at the table, NIH, FDA, CMS, HHS, to solve this problem. But our children are not benefiting from the revolution that science has delivered.
Thank you so much for those comments. Secretary Kennedy, one of the people you spoke to this morning, baby KJ, one of the ways in which that gene editing was delivered was a lipid nanoparticle, which allows the researchers to give repeat doses without the body developing autoimmunity. So if the first dose is too low and doesn't work, they can increase the dose till it starts to work. And that has been a challenge for prior delivery systems of gene therapy and gene editing. And since we have the gentleman who actually designed it, I thought it might be nice for him to directly tell you about that, Secretary.
Thank you, Dr. Prasad. Folks, I'm humbled to represent a remarkable team at CHOP, and Dr. Arns Nicholas, whom you spoke with this morning, Mr. Secretary, is the lead. Dr. Kiran Musunuru of Penn Medicine, folks at Danaher Corporation, a leading American company providing CRISPR clinical grade, and then the Innovative Genomics Institute founded by Jennifer Doudna, the Nobel winner for CRISPR where I work. And my one word to you is thank you. Thank you for making the time to speak with the cell and gene therapy community. Mr. Secretary, I loved the way you said CRISPR gives the gift of life. That's exactly what just happened with baby KJ. I will take it with your permission and quote you in all my slides. Slide one. But the thing I want to emphasize is how much promise this technology has specifically to lift American biomedicine and American health. As you look around this table, every institution represented, Duke, St. Jude, USC, UCLA, Penn, Stanford, Harvard, UMass, UC Berkeley. There's so much innovation on the way to help treatments for the next baby KJ or the third one or the fourth one be scalable and more affordable. In other words, Dr. Prasad introduced this concept of a parachute outcome, where you don't really need to randomize a trial whether parachutes saves the lives of people jumping out of airplanes. They just do. So looking ahead, I'm so grateful that the entire stream of HHS leadership is here, because I think we need a vertically integrated approach to the future of CRISPR on demand, genetic therapies on demand for severely ill children, where we start with support for the NIH. Dr. Bhattacharya, I mentioned that the NIH provided key support for baby KJ. And then we move on to CBER review, the Office of Therapeutic Products, led by Nicole Verdun at the CBER, was essential for baby KJ. And then moving all the way up the chain to Dr. Makary and Dr. Oz and Mr. Secretary, we can give American children, like baby KJ, a remarkable future free of the curse of their genetic disease. And this would be a uniquely made in America solution. I'm very serious about this. So I think now is the time to forward integrate this remarkable firepower we have at the leadership towards a fundamentally made in America solution for how genetic therapies on demand are scalable and affordable. Thank you so much for taking the time.
I know at the end of this panel, the Secretariat would like us all to get a photo together to commemorate the event. But the last speaker I want to bring up, Michelle Rangarajan, made a point that I think complements some of the perspectives that have been said here. We've talked about sort of the curative potential of sickle cell treatment. We've talked about the remarkable lessons learned from your bespoke gene therapy, a gene therapy made for just one baby, not really able to be scaled to other people. We talk about these cures and transformations, but Dr. Rangarajan made an additional point, which is that we also need to be receptive to modest step forwards as well. It's not just about cures, step forwards matter. I wonder if you might talk through what it means for a Duchenne's patient to retain functional ability just a little bit further.
Thank you so much, Dr. Prasad. And again, thank you all so much for being here. I think the example I used is the ability for a young man in a wheelchair to lift his arms and type on a computer. And in the era we live in today, maintaining that ability enables someone to independently interact with the world, to have a job, to interact with friends in just a fundamentally different way than if he needed help to do those things. And I think as we talk about cures, we also need to think about people who are alive today and living with these diseases and actually have the potential for significantly prolonged quality and quantity of life. And we can't forget about therapies that prolong quality and quantity of life for kids with Duchenne, whether that's being able to climb up the stairs at a playground and join a friend on a slide, or being able to play video games with a group of friends. It's a truly social interaction. We need to be thinking about these quality of life improvements as well and maintaining those for all of the patients that we're talking about.
Thank you so much. And with that, I think I'd like to thank once again everyone for attending this panel. Secretary Kennedy, Dr. Oz, Marty Makary, Jay Bhattacharya, and all of the panelists who contributed your invaluable thoughts and advice. We've been taking notes, we're recording it, we're gonna go through this and try to incorporate suggestions, little and big. Now I think it's a great opportunity for you all to mingle and talk amongst ourselves and directly speak to the people up here. We can get a photo together and then we can continue these conversations in the next few minutes. So thank you so much for doing this.